There was and I will ask my colleague Satifakara to share the YouTube link, just so that whoever is unable to access the room can use that. And then we can proceed. Are we good to go now, Prashant? All right. All right. I think we can get started here. I wanted to begin this conversation today by asking our colleagues just to imagine the reasons that gene drives have become so important to us. And it's really just a reflection for all of us here. And I want to run this by you guys just to see if we share in this belief here. In my opinion, at least, I think that given the socioeconomic challenges that we have in malaria endemic countries, especially in Africa, and given the weaknesses in our health systems, that many of the technologies that we have at the moment to control malaria are not going to do much, even if they were super efficacious. And this is just because we will need a lot more resources to also strengthen our health systems. And so I get excited about gene drives in particular, because I believe that they are the only tool in our arsenal today that could actually continue to work regardless of these challenges. And I just want to run this by you guys to check whether this is something that all of you believe as well. And maybe we can start with Austin, whether this is something that that you think is true, and whether your own objectives around gene drive are aligned to this. And back to you.
There we go. Yeah, very happy to be here. Yeah, so I think it's absolutely true that gene drive could have a very, very, very substantial impact on the malaria burden. We always are careful. We don't want to call it a magic bullet. We're going to need all the tools in our toolbox, but certainly gene drive can make a dramatic difference, at least in principle. And of course, it's up to the developers to actually see if that potential can be realized.
Right.
But there is, I think there's a lot of promise, as you say.
Where do you think the gaps are right now that could get us to achieve that? Of course, I understand why you don't want to call it a magic bullet. I don't think we have any anyway, but what do you think the gaps are?
In terms of getting gene drive out to the field?
Yeah.
Yeah, so I think, well, dealing with a lot of safety obviously has to be paramount.
Right.
And so just making sure, that we're not going to be doing anything with any unintended consequences. Resistance, well, anytime you do anything, you should have to worry about resistance being an issue.
Right.
And so I want to make sure that it's going to be sustainable, the interventions. And so it lasts a long time. And so I think these are the main, and there's a fair bit of, what shall I call it? It's quite a new technology, obviously. So it's just time taken to get people used to it. And that's people of in all the different stakeholders involved in this, and being involved in whether it should go ahead or not, making decisions about it. It just takes time to educate and get everybody comfortable with that this is going to be something for the good.
Great. And today we will speak a lot about all these challenges as well, issues such as resistance, both on the drive itself, but also insecticide resistance and all these other challenges that you've talked about. I think that gives us a fairly good start. And I would love to invite some of you. I know that many people who are attending the call today or the masterclass today do not have a background with gene drives or are not really conversant with the technology. So perhaps it makes, I believe it makes for a very good start that we have a very basic description of what this technology is. And I've requested Tony and the rest of us really to help us with this. So Tony, if you could just spend a minute or two to describe what gene drives are really, and how they can be incorporated and why they work against malaria.
Yeah, I'll do my best.
Go ahead, please.
I see you have a slide there, which is one form of gene drive, which is called a homing-based gene drive, and it uses the CRISPR element in order to copy itself. So the basic definition of a gene drive is any genetic element that's able to bias its inheritance from generation to generation. And through biasing their inheritance, much more than you would see the inheritance of a normal genetic trait, that genetic trait that's associated with the gene drive is able to spread into a population very rapidly. And that's what makes it transformative, because it spreads if we're talking about mosquitoes, as the mosquitoes mate, the trait spreads, and it spreads only within that population in which the mosquitoes mate, which is a single species usually. So they're transformative for that reason. And the diagram you have there shows, as I said, one of these homing-based gene drives where you have a genetic element that's based on a CRISPR unit in this case, and it's inserted on one chromosome. But when the mosquito makes its offspring, prior to making its offspring, in which one chromosome or the other would be inherited, the gene drive copies itself to both copies of the chromosome prior to those gametes being made, so prior to either the sperm or the eggs being made. And so regardless of which chromosome they inherit, it has a copy of the gene drive. And that's the basic mechanism by which this class of gene drives increase their frequency from generation to generation.
So you're saying that this is just one class of gene drives. I'm wondering whether you want to talk a little bit more about the other classes. I don't know
not
if I have
really, to be honest, because
good
this is the most,
for you, but
this is the most advanced. This is the most advanced type of gene drive in terms of getting it close to fruition. But there are other. The details don't really matter too much. The basic principle is this idea of the genetic element doing something that biases its inheritance among the sperm or the eggs that go on to form viable offspring.
Right. So would it be right to say then that if a male organism were passing a gene to the offspring, there would be a 50% chance that offspring gets that gene?
So normally, for a normal genetic trait,
for a normal genetic trait, what we refer to as the Mendelian fashion. Exactly. And you're saying that if you then add gene drive system, that process goes much faster. Yeah,
so that could be something more than 50%. In many cases, with some of the examples we built, it'd be close to 100%. So
does that happen in one generation or multiple generations, Tony?
It will happen each generation that has the gene drive will perform similarly. So what it means is that initially the gene drive
I think we lost Tony a little bit. Is that it?
I think so.
Someone wants to pick that up? George, do you want to help us here? We lost Tony.
I'm not sure, Frederick. Hi, by the way.
Hi, how are you? Nice to see you. It's really just a lot of people on the call today have no experience with gene drives, and we just wanted to spend the first few minutes to explain what the technology is. And Tony already explained the basics. Perhaps you could help us just describe what the different forms are. Put up a slide there, which is self-explanatory. You could use that to explain that.
Yeah, maybe I could, although possibly someone else could help us as well.
Shall I have a go if you want?
Okay,
go ahead.
So, yeah. So as Tony was mentioning, normally if you have if a male or a female has one copy of a gene and reproduces, it passes on that gene 50% of the time. Whereas with gene drive, it manages to get copied over to both chromosomes before it reproduces, and so it gets to close to 100% of the progeny. And so if you introduce the gene, let's say, into a population at 1%, instead of staying at 1% of the population generation after generation, it'll instead go from 1% to 2%, and then 4 and 8 and 16, 32%. So it just spreads exponentially through the population in that way. And so that's the basis of the fact that you can introduce a gene at a very low rate, and then over successive generations. So it's not an immediate effect, but over successive generations, it'll become more and more frequent in the population and take over. And so mosquitoes, let's say a generation time of about a month, so a year, that's 12 generations, you can get up in frequency very high. And so it's something that can happen in a timescale that's useful for public health interventions.
Right. Thank you.
Yeah.
Go ahead.
So on this slide, there's over on the right there, over on the left, so the dominant and most advanced sort of gene drive uses enzymes that recognize and cut specific sequences of DNA. And the architecture for doing that is to use this CRISPR system. It's a naturally occurring system in bacteria that they use to protect themselves against viruses. And what the molecular biologists are doing are taking this system from bacteria and putting it into the mosquitoes, and not to protect the mosquito, but in order to engineer this gene drive system so that you get this doubling every generation over successive generations. And there's over on the right, there's kind of two dominant ways of being able to use gene drive that are both of which are being actively pursued in the research community. One is to use them to reduce the numbers of mosquitoes of the thing you're targeting. And so the idea there, that's the population suppression. So you would introduce something, the red there, and it increases in frequency. And as it does so, it makes the population crash. And it can do that if it interferes with the reproduction of the mosquito that it happens to be in. So for example, if it makes homozygous, so females that inherited the gene from both their parents, if those females are sterile, then you can gradually suppress the population over time.
You have described previously, Austin, a number of ways in which gene drives can help control malaria. We picked up this specific image here or graphic here from one of our previous publications. Would you mind just using this to explain how gene drive helps us to control malaria, how it interrupts malaria transmission?
Yeah, so that equation on the right, that is the R0 is the reproductive number for a disease, a vector-borne disease. So it's malaria. And so this is just the mathematical formulation of all the different components that make up the reproductive number for a vector-borne disease. And that reproductive number is if an individual in a naive population is infected, on average, how many people get infected from that person before they themselves are cured or die, before that infection disappears from that person, how many descendant infections are there? And if that number is greater than 1, then that means the disease can spread through the population. And if the number is less than 1, it'll slowly disappear from the population. So that's kind of the thing that we want to be reducing when we're trying to reduce malaria transmission. It's just a way of measuring or quantifying that. So the different letters there over on the right of the equal sign are just the different components that make up that reproductive number. So A is the human biting rate, how often people get bit. Then there's B and C, that's the transmission of, I always get mixed up which way is which, but one is the transmission. If there is a bite, what's the probability of an infection going from the person to the mosquito, and the other one's from an infectious mosquito to the person. So then there's theta, that's theta E, that's the probability of the mosquito living through the period, because as soon as a mosquito gets bit, bites an infected person, that mosquito is not immediately infectious. The parasite has to develop within the mosquito. So that's about 10 days, or give or take. I think other people can. And so it's probably that the mosquito lives through that incubation period of the parasite within the mosquito.
And this is the information that we need to calculate the
Yes. So
likelihood of one.
And we can try and change any one of these numbers. Mm-hmm. So mu A is the daily survival rate of the mosquito once they're infectious. R is the recovery rate of the human. That's about the only thing on the that we're not affecting here. That's how what's the probability, the rate at which mosquitoes will recover from the disease. And then big A is the number of female mosquitoes per adult human being.
Right.
And so any one of these numbers or letters, parameters, is something that could be if we could target it with a gene drive, would have the effect of reducing the R naught. So the population suppression approach is reducing the numbers of female mosquitoes, the big A. Mm-hmm. And the population replacement approach, where you're leaving numbers intact, but you're trying to reduce the probability that the that the parasite can develop through the mosquito, that would be reducing the B or C parameters.
Thank you. Thank you so much, Austin, for this. We have at the moment, and I think it's important for me and my colleagues to say a big thank you to the teams currently working on gene drives around the world. I mean, I do not know exactly how many they are, but the three main ones that we have here at Target Malaria, we have the group at University of California, Transmission Group, Zero Group at Imperial, and I think there are several other teams. So we my apologies if we didn't include the exact name there, but we are grateful for all this effort that the different teams are putting in. We have one question that we will come back to later about the combinations of the different forms of gene drive. But to begin with, I would like to invite my colleagues working with the Transmission Zero project at Imperial to kind of just give us a little bit of a highlight of what they're doing and some of the progress that they have had in the recent weeks and months. So I don't know who in the team would like to talk about that, but either George or Nikki or one of you guys, or Dixon, my colleague at Ifakara, or Tib, one of you guys, if you want to just tell us a little bit about Transmission Zero program and this exciting news that you have released in the past month.
Yeah, I mean, I could go ahead, Fredros, and our colleagues in Ifakara could possibly complement what I'm saying. So Transmission Zero was set up as a population replacement program. So Austin and Tony explained previously the different types. So Transmission Zero is a population replacement gene drive program. So what we're trying to do essentially is to make mosquitoes not being able to transmit their disease. And how do we do that? Is to one way to do that that we have made significant progress at the moment is to make the mosquitoes immune to the parasite, so they cannot contract the disease, or if they do, they will be able to kill the parasite before it is mature enough to be transmitted from the mosquito to people, before the mosquito bites again, essentially, to transmit the parasite. So what you show here, so along that way, we generated several tools. We had to innovate, we had to build capacity to do that. And along that path, we have the what you show here on this slide, which I believe is the first transgenic mosquito that was ever generated in Africa, that was generated by our team, by our colleagues at the Ifakara Health Institute. And this mosquito is not really the one that we want to experiment with in the future with blocking the parasite, but it is an intermediate tool. So you can see that this mosquito, which is a transgenic mosquito, again generated in Ifakara, is fluorescing, has this red fluorescent, which can tell us is to distinguish between these and the other mosquitoes that we have in the lab. And it tells us that this is a GM mosquito. So that was one of the innovations, for example, that we generated along the way. Now, in the laboratory, we have another great innovation that that we feel that we might be in position in a few years to test whether it can have any impact in the field, which is indeed a mosquito that cannot transmit the parasite. So it kills the parasites as soon as they come in. It kills all the parasites. Actually, it doesn't kill them. It delays their development. If I take it back to the reproductive rate, the disease reproductive rate that Austin very nicely explained, these mosquitoes affect the time that the parasite needs to be within the mosquitoes in order to be transmitted. So the parasites are very slow in that mosquito. They don't mature very quickly or as quick as possible for them to be able to outcompete the survival time of the mosquito. So by the time the parasites are mature enough, the mosquito has died, so it won't be able to transmit them. So that's one of the things that we are targeting, for example, here. Yeah, so Transmission Zero is a program that for the moment consists of 2 partner institutions, and this is Imperial College, where we develop the technologies, and the Ifakara Health Institute, where we validate the technologies and we develop the innovations that would be applied in the field at the end.
And of course, from Tanzania side, there's also the partnership with the
Yes,
National
of course,
Institute.
I totally forgot. Sorry, and apologies for my colleagues at NIMRI. The Tanzania National Institute for Medical Research is also a partner in this project, and they take on the field component, the one that is closer to the communities in Tanzania.
Thank you so much, George. I'm wondering whether any of your team members would like to participate, to contribute as well. I can hear a voice. Is that Dixon?
Yes.
Go ahead.
Thank you very much. Thank you very much, Freda. Thank you very much, George. So I guess the other aspect, apart from the technology development, I think is enabling environment, or now we can validate the hypothesis we have that this mosquito can block the transmission. And I think within that landscape, there's different steps that we've been part of as Tanzania championing the development of this tech. And what I mean by that is, first, we should acknowledge that validating the development of this tech require investment in terms of the infrastructure. And it's not about just putting the lab. It's a question of you install it, you make sure you sustain that biosafety required measures. And the another aspect is building technical skills. I know we have a critical mass in the Western that people understand this technology, but I think it's quite new on the southern part of this space, and therefore it's very important for people to understand and learn the technical skills that are required toward development. The other aspect, which is extremely critical, is also starting engaging the all relevant stakeholders, which have been heavily involved, would not only give us a ticket for acceptance and implementation of this tech, but also generating the knowledge and the awareness. And the reason I'm saying knowledge and awareness, this is quite new. And I think it deserves a co-production of knowledge, so people knows, even when I'm not around, the friends are not around, but there is people within the public domain who understand what's important of this tool and what can it do in terms of malaria elimination and eradication in that regard. So if ICARA is heavily involved in that line with Imperial supporting the development of technology, but also putting all the infrastructure required toward validating that technology, but also engaging all relevant stakeholders that will be supporting the acceptance and its implementation and building local capacity that is required to sustain the technology on the ground. So that I wanted to highlight those few bits. Thanks. Back to you, Frederick.
No, thanks a lot. Thank you so much, George and Dixon. I think it's important really that you guys highlight the successes that you've had here, and we've had great successes also from the Target Malaria team and also from the University of California team. And I think it's all this sometimes go unnoticed. And on behalf of my colleagues, of course, again from the malaria endemic countryside, we just want to say a big thank you to all of you for the efforts that you do. At this stage, we would like to bring in our colleagues joining from endemic countries, or experts from the endemic countries, to talk to us a little bit about what gene drives mean in terms of malaria control. So at the moment, of course, we are mostly using insecticides, nets, and drugs, and diagnostics, of course. And we think that gene drives has got very good potential that it might make its way into malaria control strategies. And it would be nice to hear very broadly what people from the countries think the role of gene drive might be. So please jump in and talk to us about this. Damaris, our colleagues from NMCP Nigeria, if you're there, Dr. Shija, Zanzibar, Frank Chaki, Tanzania, if you guys are there, please chime in. Go ahead.
So maybe I could start, and thanks, Fredros, for inviting me and others to this and having an opportunity to share our thoughts and experiences around gene drive. So for me, from the research component, I think this is a great initiative. And as we started, we can't say it's a silver bullet. I wish it was, because if it was, then we'll put all the energies and resources to it to ensure that we achieve control. Because one of our Vision 2030 is malaria elimination or eradication from various, at least 35 countries in the continent. So having interacted with various scientists, the policymakers, community health workers, and the community in general, I think one of the perceptions and thoughts we've seen over time is that the communities are desperate to get tools that can actually control or eliminate malaria. Discussing with women at the community levels, at household levels, they were like, please, can you bring for us this tool if it's going to reduce malaria, because the burden is too high, having to deal with children who are sick and the families that are suffering from the disease. But talking to scientists, on the other hand, the reservation is like, we are not sure. We are not sure the effect it will have. We are not sure whether these vectors are going to result into something else. When we talk to the environmentalists, they are talking about what impact will it have in the ecosystem, especially maybe having hybrids of mosquitoes that will impact the environment differently. So the ecosystem is a question. But talking to policymakers, for them, they are open and thinking that you scientists tell us whether it's working, and if it's working, how do we incorporate it in our management, disease management algorithms to ensure that it's rolled out and bring malaria down substantially. So from my end, I'm thinking that it's important to advocate and ensure that this tool is also, as we've done previously in insecticide-treated nets, as we've done in the use of IRS, larval source management, and also case management, even including therapeutics. So this being an innovation, then it's important for us to ensure that everyone is on board, starting from the key stakeholders, yes, the researchers, but also the regulators, that's the National Biosafety Control Authorities, the policymakers, and also the communities to come and start the discussion. Coming from Kenya, where we haven't much, have not had much, we are not privileged as Tanzanian, where they have started this technology. We are learning from what Burkina Faso is doing. We are learning from what Tanzania is now doing. We are learning from various other institutions, what they're undertaking. So for us, the desire is to ensure that we learn from the successes that they have had so far, and also use the experiences to roll out this tool in the Kenyan population. So I will stop there, but I think the tool comes in time when we desperately need control measures, and also to counter the resistance, because there's a lot of insecticide resistance that has been reported so far. And as you know that in Kenya, we are using a lot of pyrethroid tools, such as insecticide-treated nets that are treated with pyrethroids. And now we are seeing resistance. Now they're coming up with the new generation nets, use of PBO related. And this also soon we might be declaring them obsolete, and they're not working. So if this tool is going to push us towards elimination, then it's important for us not only just to start the discussion, but to ensure that it's rolled out with everyone owning it. Thank you.
Damaris, I know you want to stop, but a quick question here. Can you confirm what you're saying is that on one hand, you have the scientists who are very doubtful. On the other hand, you have the potential end users who are very optimistic. Is that the case here?
Is the case, and one of the things, because the scientists that we've had discussion with, they are broad, they are range of from the public health research scientists to the lab techs, and to also people who are involved in assessing even mosquito surveillance and etc. And most of them, I think, one is lack of information, so that's why discussions like this are important. But to demystify that, they are the doubts they have, to tell them how the tool works and how it's going to counter the doubts that they have. So, but the communities, them like, tell us what is working, because we are tired of this disease. And also maybe, Fredros, back to you, learning from what has happened in other countries, not in the African continent, but in the US, in other places where malaria was previously. How can we bring malaria reduction using, yes, gene drive, but other innovative measures that can help us bring this disease to control, and come from 96% of the disease occurring in the African continent to lower levels that are manageable. Yeah.
No, thank you so much, Damaris. This is a very important topic. So in case we have other members from other countries who would like to contribute, please feel welcome. Just let me know and jump in. And before we move forward, I just want to check whether my colleague Lena has any follow-up question here. Yeah? No? All right. Okay. So let's move on a little bit more. A quick question to our panelists in here, and also our colleagues from the countries. I think Damaris was mentioning this already, that we have, other than gene drives, we have a number of technologies that are in the horizon, that are being developed, that are almost equally exciting, if you like, depending on who you ask. And I mean, it would be nice to know. And you can put your responses to the chat as well, if you like. It would be nice to hear from people in the broader room as well. How do you think this is going to fare? Of course, you talked about gene drives alongside the interventions that we currently have. You talked about new generation bed nets, but what do you think is going to happen when we start to have these new tools in place, attractive targeted sugar baits, monoclonal antibodies, potentially new vaccines? What do you guys think is going to happen here? So I would like to throw this question to any of the panelists who might be interested in responding to this as well. Hey, Richard, are you trying to speak? Is that Professor Mkabana? Anybody has any comments around this? How gene drives will match up against these new interventions?
Friend Ross, let me go.
Go ahead, Prof. Thank you so much.
Yes, I think one of the things that we really need to think about is how are we going to integrate all these interventions and making sure that they all work in tandem with each other. In other words, are they going to work in synergies or are they going to destroy each other? So we really need to find ways and means of integrating, because yes, suppose we come and say gene drive is going to be the main one and we release it, yet we are doing indoor residual spraying, we also have the nets, and then we are also saying, okay, we also bring in other tools like vermectin or ATBS-B and so on. So how best are we going to integrate these tools? So that is the key, and we need to work to have a group really working on this so that we can be able to maximize the benefits of each one of them. That's my contribution. Thank you.
Thank you. Thank you so much. Thank you a lot, Prof. I'm just wondering whether this is a question that will need field data or if it's something that we can do predictions on based with mathematical models, for example. My colleague Prashant is on the line. Prashant, what do you think is going to happen here? Is this something that must wait for field data as well, or is it one of those questions that you could at least test out with simulate in mathematical models?
Yeah, I think field data is always gold standard here when you're dealing with models. So if field data is available, that is, you can't beat that. But for some of these interventions, there are trials ongoing or they have been completed, like with ATSBs, monoclonal antibodies. There's some trials going on right now. Vaccines, we've had trials. So we have data from that. And using that data and combining it with like gene drive models, we're seeing that it is a synergistic effect. Even with things where you would think it's not intuitive, right? So with ATSBs, for example, you could be killing off the mosquitoes, but you still have a synergistic effect, right? So you release these gene drive mosquitoes and you could potentially kill them with the ATSBs. And so you think that they won't work together. But what we're seeing is that even with these vector control tools, we get a synergistic effect. And with the human-based interventions, so like monoclonal antibodies or vaccines, it's a completely different pathway, right? So the chances of that being synergistic are higher. And so we're seeing that usually it's kind of nice to have this co-deployment because, you know, if you're missing out, it's kind of like this the Swiss cheese model, I guess. If you're missing out with one of the interventions, then hopefully the others will clean it up.
Yeah.
And so we're generally seeing a synergistic effect, but I imagine like in some cases, especially with vector control and gene drives, you could see some kind of you know, like a kind of a control mechanism on the gene drives, where you where the gene drives are not just spreading on their own, and you could use like an ATSB or a net or something like that to kind of stop them from interacting with humans.
So let's go back to this BedNet and IRS question here, and either you, Prashant, can answer, or we could go back to Tony and Nikki here to ask. But you bring up an interesting point here, which is even though we expect these things to be antagonistic, if you like, that they're actually not. Does this mean that the gene drive candidates that we have, the gene drive products that we have at the moment, are low threshold enough that they will continue to work even if they are deployed in the same areas where you already have killing agents like IRS or ITNs?
Yeah, I think I would want someone else working on the drives to answer this, but mathematically speaking, it does depend on what kind of drive you're releasing. If you have a low threshold drive, I think one of George's responses in the chat was about how the super Mendelian inheritance can be used to overcome fitness costs. And you could think of interventions as a fitness cost, right? Trying to kind of stop the gene drive as well. So it depends on the kind of gene drive you're using. If George or someone else wants to step in. Austin, perhaps.
Yeah, please, gene drive developers. What do you guys think? Should countries worry about, go ahead, Tony, worry about combining ITNs and IRS with gene drives, or is this something that is a no issue? Go ahead, Tony.
Well, I don't want to speak over the models, and I couldn't quite hear all of what Prashant said, but I think in most cases, it may be counterintuitive for many to think that, or to realize that, in most cases, they are compatible regardless, or in most cases, in the worst case, they'll be neutral, I think. Because, if the thing is invasive and designed to invade a population, if that population is reduced, there's less of them to invade. The counter side to that is that more of the gene drive mosquitoes will be reduced, but they're still invasive to the remaining population. I think that would be the main message. Although I see on the chat, and it's something I wanted to raise, someone made the point of testing them in isolation, because one of the things that they confound each other on is the actual magnitude of effect. If you have 2 or 3 different approaches that are all with the same goal of reducing a vector population, then attributing the reduction in population to any one of those interventions can be tricky. So I think that's something that needs a bit of consideration as well.
Wow. Thanks a lot. That's an important point, given that these are currently our most important technologies, and you want to be at least reasonably confident that you are synergizing, not compromising the existing interventions. We're going to go into a little bit of a technical deep dive here, but I hope that you guys can help us make it simpler. So Nikki, are you there? I would like to invite you to the podium, if that's okay. Are you there, Nikki? Yeah. Good morning, Nikki. George, do you know if Nikki is on the call?
I don't think he's on the call, Fredros.
Okay, so maybe you and Austin and Tony, if you're still on the line, you guys can help us with this. We were just trying to understand a little bit about the history of this technology, the recent history of this technology. We find the year 2015 to be a particularly exciting one. And often when the last time we see in history things like this happen was during the human genome competition between the UK and the US scientists. And in 2015, you start to have, first of all, this fantastic written paper from the US, fantastic summary there, the easiest abstract to understand, but using the term mutagenic chain reactions. And right after that, you have the publication from Imperial College and a follow-up publication from the US again on that, with a fairly large payload there, nonetheless still very efficient gene drive constructs. And it would be nice to just hear from you guys about this period, this time in the campaign towards gene drive constructs. What was it like? What did all these things just all of a sudden come to light? What was the trigger point? How come everything was just coming out? Because in 2014 you already have a nice work from Imperial, and then in 2015 the CRISPR comes in and everybody is churning out these beautiful pieces of work. What was it like at that time, and what really made this takeoff happen? Hey, you're muted, George.
Yeah, no, I was just saying that maybe we should let Tony explain that, because he was part of this development at that time.
Yeah, Tony, go ahead, please.
That's kind of you, George. Yeah, so, well, let me go back a little bit. So it was Austin actually who had the first idea really of repurposing this type of selfish genetic element, which is based on this cutting mechanism. that gets it from one chromosome to the other. Without boring you with the details, those are called homing endonuclease genes. And they have a very specific recognition sequence in those organisms that they invade, which are usually relatively simple single-celled organisms such as yeast and some other fungi. And they recognize that sequence, they copy across from one chromosome to the other. So the idea was, could we make that happen in the mosquito germline prior to it making its sperm and egg, and have a similar invasive element that would be controlled as to where it cut in the genome, where it located, and spread through a population. So that was the basic idea, and that didn't really change and hasn't really changed. It's the actual tool with which you can build that type of genetic element. So a way to specifically recognize a sequence that's in the mosquito genome. And prior to the advent of CRISPR, what we had to do was take these existing homing endonuclease genes and try and understand what it was in their protein structure that allowed them to recognize a specific DNA sequence in the yeast genome or whatever it was, and try and understand that protein interaction with DNA and re-engineer it so that it would recognize a site in the mosquito genome. And then on top of that, you have to make it active in the cells that you want, all the rest of it. But those things are relatively manageable. We could already do those. What we didn't have was a way to efficiently reprogram, redesign these endonuclease genes to recognize sites in the mosquito genome. So we had some ways to do it, but they were long, laborious, a lot of trial and error, and difficult to make work. And you need to make them design regions in mosquito genes that have the desired effect when you disrupt them. So they might be involved in female reproduction, they might be involved in parasite permissiveness, or some other interesting trait. So you have a limited set of sequences that you can actually target, which makes it all very difficult. Now, of course, when CRISPR came along, the whole thing changed, because the CRISPR element is essentially very similar in that it's a DNA cutting enzyme. And so you can repackage it to make it work like a gene drive. And with CRISPR, in order to recognize a particular sequence, all you have to do practically is provide that sequence in the form of what's called a guide RNA, which allows that CRISPR-Cas9 element to go and target that sequence in the actual genome that you're targeting. And so once that happened, the door was open to basically make these elements that we'd already shown proof of principle for in the mosquito germline, make them now with CRISPR. And that was the transformative step that made it all a lot easier. Does that make sense?
Well, that makes sense for me. If someone has any questions here, I guess for me the only question there, Tony, and forgive us for sticking to the technical detail here. The work you guys published in 2014. This was pre-CRISPR, right? So this is the example that you did before.
So this showed you that if you were to hook up a nuclease and make it active in the mosquito germline, would it copy? And the answer was yes, it will copy with high efficiency, so it will be inherited amongst most of the offspring. But at that stage, it was just using a sort of a dummy sequence. It wasn't targeting anything endogenous, really, of interest in the mosquito genome. Okay,
thanks a lot. So let's use that example to explain something a little bit important here, which I think many partners today on the call would like to understand. This is the question of fitness costs. At the time of all this series of publications, our colleague Luke Alfie published this editorial where he tried to to explain the differences in the fitness costs between the replacement drives and the suppression drive. I'm wondering whether one of you guys can explain a little bit how different these two are in terms of their ability to initiate resistance, for example, and their ability to survive in the field against the wild.
I can carry on.
Yeah, go ahead.
Yeah, so a genetic element that's copying itself, there are many genetic elements that copy themselves in genomes. And as they do so, they impose a fitness cost on the host organism because they are effectively genomic parasites. And in the same way, a virus replicates itself in you, or a parasite replicates itself in you, it imposes a fitness cost on you as it replicates itself. So that's not something unique to gene drives. Now, a gene drive that is affecting a mosquito's ability to reproduce and thereby suppressing the population is imposing that fitness cost directly ultimately on those mosquitoes that have that genetic element.
Yeah.
One that's doing population replacement may or may not have a direct fitness cost on the mosquito, but it will certainly have a direct fitness cost on the parasite. So it will place selection to be resistant to it on the parasite rather than the mosquito. So that's where the sort of main differences lie.
So is that it?
Yeah.
Yeah. So let me. The reason I ask this is because I think it helps us to lay a basis here for an important difference between the two kinds of gene drives. We could come back to that at a later time, but this is the question of resistance to the drives. Before that, though, is it true at the moment that the most advanced gene drive product that you guys have is the so-called double sex? Is that right? Austin, is that correct?
So yes, so the target gene that we have been focusing on most recently.
Yeah.
So for the population suppression,
yes,
the strategy that's being adopted is to knock out to use the gene drive system to disrupt or knock out one of the endogenous naturally occurring genes of the mosquito, and a gene that's involved in female fertility, such that if the females are homozygous, then for the knockout, then they can no longer reproduce. And so the target gene, which we have settled on most recently, is called double sex. And it's a gene that's involved in the sex determination pathway of the mosquito. So the pathway that determines whether a mosquito develops as a male or a female. And the essential feature of it for our purposes is that females that are homozygous for a knockout of a particular region of this gene are sterile and so cannot reproduce. And as a bonus, they're also aren't able to, they don't have the right mouth parts, so they're not able to bite and transmit the disease. That's not an essential feature of it, but that's kind of a nice bonus to have there as well, that the homozygous females are unable to transmit.
So just hold it there. So essentially, the genetically modified mosquito that Target Malaria is proposing to use is the one that results in this, what shall we call it? This mosquito that is part male, part female, right? So the image we have on the screen here.
Yes. So this is what homozygous females,
right,
have what's known technically as an intersex phenotype.
Right.
But what matters is that they're sterile.
Excellent.
They're unable to reproduce, and so if you can make lots of homozygous females, then the population will go down, will be suppressed.
Okay, but when they are in the field, and I'm asking this because of the monitoring question. So if I was an entomologist and I'm looking for, I'm trying to figure out whether the suppression drive is spreading in the field, it would be sufficient to look for the mosquito that has both male and female body parts.
So our intention is to be looking using molecular approaches to look for the gene drive in the DNA, because if you're a heterozygote,
you won't have that.
You won't have any phenotype, and if you're male, you don't have any phenotype. So the actual ones showing the phenotype are going to be quite rare in the population, and we don't know what their survival is going to be in the field. So we would not rely on looking for those. Instead, we would rely on doing PCR, looking at the DNA to see which, both males and females, to see which ones of them are carrying our gene drive construct.
Yeah, an important question here was whether, I mean, if you're going to trap these mosquitoes, you want to be sure, can these mosquitoes even come into a host-seeking trap?
Exactly. So we would not be relying on, yeah,
okay.
Exactly.
All right, so your suggestion then is for monitoring purposes, endemic countries should be building towards more molecular tools to monitor gene drives instead of just relying on the morphological features of the double sex, even though some of them, the homozygous females, homozygous ones are transgender mosquitoes, if you like.
Absolutely, we should be relying on developing up the capacity for molecular surveillance for the gene drive construct. Yes.
Okay, thank you so much for the clarification. Tony, go ahead, please.
Yeah, that's an important point. I just wanted to put in there, Fredros, when you say, about the molecular screening, it's important to mention that, molecular screening gives you a very categoric result. It's very unambiguous. But it's also important to say that we don't need to reinvent the wheel for this, nor make absolutely new capacity, because, also for insecticide resistance, molecular screening is taking hold now because of the unambiguous answers it can give you about the genetic origin of resistance. So in the future, there are a lot of moves to basically integrate the two types of capacity building, so that while building that capacity building around screening for insecticide resistance is the same type of thing that's necessary for gene drive. So it's not like it's a particular ask or requirement of gene drive.
No, thanks a lot, Tony. And at this point, I would like to actually throw it back to our colleagues from the countries just to ask what they think it's going to take to roll out the level of molecular surveillance that we will need to monitor gene drives. As our developers have explained here, some of these mosquitoes will have these physical features, yes, but the more accurate way to do it is to try and trace them using a molecular approach. So I would like to invite our country partners just to help us understand to what degree can countries actually prepare for and roll out molecular surveillance techniques that will be necessary for, let's say, district-wide or country-wide assessment of gene drives, using this as an example, and later we will talk about integral gene drives as well. So any of you, any of our colleagues from Nigeria, Tanzania, Kenya, Mozambique, please go ahead. Who is going to rescue us here? Damaris, we come back to you. Do we have a capacity? Do you think countries will be able to put up the molecular surveillance to the degree that we need to monitor gene drives at scale? And I assume here, Tony, that we are really just talking about PCR. It's not like we need sequencing, right?
Yes, I think maybe for molecular surveillance, Fredros, I think to learn from what, to say that it's a blessing in disguise that COVID came. So currently, I could say that most countries in Africa have capacity to do even basic PCR, basic molecular biology. So I believe that we have the capacity to track, because if it's continuous surveillance, which we are doing, especially like in various countries, there's continuous surveillance from the sentinel sites where we are tracking occurrence and spread of the malaria vectors. So we could use this, the expertise we have, like we have currently trained district entomologists to even just trap and preserve the samples and send them to the various reference laboratories to just screen the vectors and tell us species-wise, but also maybe now what we might need from Tony and the team is the molecular markers that we might need to just check for presence, such as whatever drive that would be in the mosquitoes, so that we differentiate them from the normal, the wild type to the versus the ones that have the drive. So what might be needed is basic training on just screening that the drive. Otherwise, countries in Africa at the moment have the capacity to do just basic PCR, and to add to that, we have those who can do even just genome sequencing, whole genome. Most labs now have the Illumina, the next generation sequences, so that can be done.
So essentially, you're saying that with regard to molecular surveillance for gene drive technologies, we are already sufficiently, or at least we are gearing towards sufficient preparation to be able to do that, right? Okay.
Yes, we are,
that makes me very happy.
Both for laboratories and also maybe just building capacity for human resource. Yeah, thanks.
Excellent. No, excellent. This is good. I would like to assume this is true in most countries already. I saw Tony raised his hand. Someone else raised their hand as well. Let me see who that is. Tony and George, go ahead.
Yeah, so you asked if basic PCR would suffice, and the answer is yes for detection of the gene drive. But as with insecticide resistance, the more information you can get, the better. So you know, if you can sequence it, it's better, but it's not essential. And you know, Damaris raised a great point. The capacity is already building thanks to COVID, and I think there's definitely a notable increase in capacity at the personal level, but also at the infrastructure level, with ability to do sequencing. And you know, I have a project with UVRI in Uganda and with Burkina about integrating some gene drive panels with insecticide resistance. So it would be basically a silent change, really.
No, that makes us all very happy. Thank you guys so much. George, and then Richard.
Yeah, no, it's just to add to what Tony said, essentially. I mean, it's actually, I would agree with Tony and disagree at the same time. I mean, the more we know, the better it is, but the more we know, the better it is for the developers. When we reach the implementation phase and the monitoring phase, I believe there's a huge space for innovation there. And this is what this community could do. I mean, the entomology community in Africa could do, or the countries that will implement these technologies. PCR might not be. I mean, it might be available to most of the places at the moment, and the technologies are there, but there will be, and there are cheaper diagnostics that could be developed that will be equivalent to just looking at it under the microscope, essentially, in terms of the feasibility. So there's a huge gap, and there is time until these technologies are implemented. There is enough time to innovate and gear towards this mass monitoring of the implementation.
But George, what you're saying is, instead of waiting, there need to be some parallel development going on right now to do that, to build up these capabilities for that.
Tools, I mean, the diagnostic kits could change in the future. I mean, based on whichever gene drive technology each country or each region will select. But the knowledge has to be passed on now and build capacity, essentially, to be able to do that fast diagnostics in the future, genomic diagnostics in the future.
Thank you. Thank you so much. Thank you. Professor Richard, go ahead, please.
Thank you, Fredros. The issue of molecular surveillance, I think, is important. And there are many labs that might be able to upgrade and start doing this kind of stuff. But for me, if these things don't get into policy, if they are not part of the policy instruments to follow up this mosquito in the field, then we're very likely to get into a problem. The people that work on GMO crops, for example, if I look at the Kenyan example, you actually have institutions which are there to regulate this, and you have people whose work is just to go, for example, the market, take some maize, and they'll tell you if they have GMO material or not. So in terms of health, I'm still wondering or thinking that we should try to look for mechanisms where what we're talking about is mainstreamed into policy, so that we are able to really do the correct thing now and into the future. I really believe that we're going to get into that. I really believe that things around gene editing are going to be the next revolution. And if they are going in the health direction, we must, know how to deal with them properly with the decision makers. Thank you.
This is very well taken up, and I do hope that our colleagues listening can take these up as well. You're absolutely right. It's going to have to be done systematically. Richard, before you drop off, I'm also wondering whether our colleague Frederick is around, Professor Frederick Tripper. Yeah. I wanted to ask both of you, maybe with Frederick, if you're there, kindly jump in a little bit. Yes, I mean, so our colleagues have told us that we need to focus more on the molecular surveillance, but you have to first trap these mosquitoes, right? And something like double sex, how do you trap this insect? Will this thing even get into a trap?
That's a good one.
Yeah.
And then there's also the issue that mosquitoes get rarefy themselves depending on the construct. So you have less and less to work with. So the timing of monitoring can be complex with some of the constructs considered. Yeah.
Right. So do we need to also be working on trapping methods, or maybe focus more on larval sampling, stuff like that?
Yeah, so that's an option. Or you increase the frequency of sampling to try to catch that short time span where there's a bit of the GM there, but the mosquitoes have not disappeared yet. So I think there are a lot of challenges, but it really depends on the strategy being considered and the dynamics of the construct. So it will have to be looked at for sure, because there are some oddities, but there are some other constructs that should be fairly easy to monitor. And if you think about the replacement strategy, possibly that's one advantage, where it's going to be easier to monitor in a sense.
Thank you.
Some people are very strong, actually, Fredros, that larval sampling might be the future, since as we are expanding to other species and other subtypes, etc. So maybe that's another area of development that we need to take.
To do, yeah. No, I agree with you. We see this with the Anopheles stephensi already, widely. Very, very briefly, Tony, if you don't mind. I see the. Oh, go ahead, Damaris.
I wanted to contribute to the molecular surveillance component, and I had a question to Richard. And here, Richard, I'm thinking, even as the teams design the tool, it's important for us to have SOPs for doing this molecular surveillance. What are the procedures, or how, even now when we talk about trapping methods, we need, so that we know that we have great methods that can capture this. However, to Richard, especially having sittings or meetings together with policymakers, so that we are able to bring them on board, so that there is also one talking, can I call it, same level of discussion between scientists who are doing this molecular who are developing, yes, the drive, but also those who've been involved in monitoring and evaluation of this, so that they are also speaking from the same front. So I'm wondering, Richard, you being at the front line, working together with policymakers, whether this is happening, or how do you plan to do this?
Unfortunately, in all spaces of SAT, that is not happening yet. And I think it's upon us as scientists in the health sector to remember to work very closely with our regulators. It's very interesting that the regulators in this space are very happy about this gene drive technology, and they embrace it very nicely. And I think that most of the doubts are really on our side, so they're very open. And I'm sure anybody approaching these people, they'll be very happy to take this up and start seeing we do everything together. I don't have enough time to pull out just one slide to show you how the gene drive technology links to other sectors, and maybe ask ourselves, are we working with people in all these areas so that we can get all this goodbye, something. Yeah, so that is something which is not happening yet, and I think we're getting to the time when this should happen, in brief. Thanks.
No, thanks a lot. Thanks a lot. And we do have a session coming up shortly on gene drive regulations as well. So maybe this comes back in. Alphan, my colleague, kindly requesting you to help me manage the room. So admit guys coming in. Let me go back to Nikki, Tony, George, Austin, one of our target malaria colleagues, if possible. A very quick question here. All right, that is not call, okay? Good. Tony, are you there?
Yep.
Tony, help us understand here. Why do we need, okay, and forgive us if we misunderstand it, but once you have the double sex, which seems to be working fairly well, what is the idea of adding the male bias sex distorter here?
Well, I mean, it's just equivalent with sort of.
Is that some kind of a double, an overkill, or what?
No, I wouldn't call it overkill, but it's equivalent of sort of hitting two pathways with a drug. If you use some sort of chemotherapy, if you can have 2 points on which the gene drive is acting to reduce the reproductive potential of the population, then you get a more severe reduction in the population, and it's less likely that you will get resistance to both mechanisms, that because it's targeting two mechanistically different pathways in achieving its goal of reproduction. But if I may, go ahead. No, go ahead. Go on. You had a question.
Our understanding from, I mean, again, forgive us for misunderstanding literature in case we do so, but our understanding from reading about double sex was that it was already fairly robust against resistance.
Sure, but it's like it's like anything. You develop an antibiotic, and it's great, and it has a predicted impact. But even the best antibiotic would be expected at a certain point.
So here you guys are just being proactive.
Select for resistant variants, and so if you had the tools at your hand that would allow you to have two different mode of actions with a single dose of antibiotic regime,
right?
You would largely do so, all things being equal, and there being plenty of other antibiotic choices. So it makes sense to investigate options that might be more potent. As simple as that, really. So in that construct, it is directly in something that's essential for the female, but it's also biasing the offspring so that there's even less females that need to be converted into double sex knockout females. So there's just a preponderance of males and a preponderance of males, and an absence of females means that you get population crash as well for that reason.
Okay. No, thanks. I saw Tony's hand, Austin's hand. Austin, did you want to add anything?
Yeah, just to add, so an additional reason early on for investigating this possible adding the sex ratio distortion was that the early version of the double sex, this is a bit technical, of the double sex construct, they had a strong fitness advantage in the heterozygous females. And so this was likely going to be necessary for it to spread. If if the fitness cost in heterozygous females is too high, then the gene drive won't spread. And so this was a way to get it to spend most of its time in males and avoid that fitness cost in females. Subsequently, we've gotten other ways now to reduce, by changing the promoter, the bit of DNA that controls when the gene drive is turned on, and so which has reduced the fitness cost in heterozygous females. And so that particular rationale has fallen away slightly.
Okay, so you're updating it even further. That's good.
There's constant update. Absolutely.
That's nice. That's nice. I think that's an important point here. I mean, one of the issues that keep coming up is, are we going to have more than one product? Are we going to have a product that is robust enough against gene drive resistance in the field? And so when you guys published double sex, it was already really, really good. And then now you have this sex distortion, which is, of course, fairly technical. It took me a while to follow, and I still don't fully follow it. But of course, someone then wonders something that's already 99% performing, and you're adding something else here. But we get the point, and I do hope that my colleagues on the line appreciate as well why the gene drive developers are trying to optimize the product as much as possible pre-release, so that they have not just a very good product, but a very, very, very good product. and
more than that, I think all the developers are all intending to have kind of a pipeline of products.
Pipeline of products, yeah.
Yeah, so it's not just one, because you got to do, it's just like an insecticide. Eventually resistance might evolve, and you want to be prepared with another construct. So, when you take some going from the lab to the field, there's steps which you might learn things there, which things don't work as well in the field as you expect. And so again, that's another reason for having a pipeline of innovation behind your lead product. Yeah.
Pipeline of innovation, pipeline of products. Thank you so much. Yeah. Thanks a lot. Tony, you have something urgent to say?
Yeah, because it was raised a couple of times, this concept of carrier individuals. So that's the idea that, individuals, that certainly females, that have one copy of the gene drive, for the gene drive to work, they need to be viable, and they need to be able to mate in order to transmit it silently to the offspring and get it into the population. So if a gene drive is working, then the number of carrier females is increasing all the time. So this idea that there can be gene drive containing mosquitoes that are hiding from sampling is a slight misunderstanding of the situation. So if they're there and it's working, then they will be detectable, at least in the carrier females, because they must be mating, they must be finding hosts, and all the rest of it.
No, thanks a lot. I am nodding as if I fully understand. I'm not sure I fully understand, but I am trying my best. This is a fairly difficult subject, but I guess you want to drive it on both sexes, so that I mean, I'm not sure though. We can discuss this offline later. But I remember having a conversation with, I believe, Stephanie. Maybe Stephanie James is on the call, if I can put her on the spot a little bit. Whether the processes that you guys are taking start to move you towards some kind of a mix of replacement and suppression in the same product. Is that the case, Tony?
Well, I think there are scenarios in which the two types of approach could be compatible and even synergistic.
Okay, yeah. we can discuss that. Let's move forward. I would like us to jump a little bit and go back to our colleagues from Transmission Zero to solve a different problem. And this is the problem that it might not be readily acceptable or readily possible for countries to test a driving mechanism. And again here, George, forgive us if we misunderstand the technicalities here, but looks like you guys have a fairly good idea around stepwise evaluation of these products in a way that allows you to build capacity in endemic countries without necessarily having the gene drive there yet, and go through the regulatory loops. And it took a while to understand the history of this, because you guys published the paper on integral gene drives, but the one that really follows your most recent work is this publication in eLife here, where you do the actual conversion. So could you just explain to us a little bit what the concept here is, how you try to go around the fact that it might not be immediately possible to test. So in Tanzania, for example, we have this transgenic mosquito, but we don't have gene drive in Tanzania. So can you talk to us a little bit about that, and maybe Dixon or my other colleagues from Tanzania can jump in as well, or any other person from Transmission Zero. Back to you, George.
Yeah. So the idea here, I would take it back possibly our colleagues here remember, you've shown one slide previously, Fredros, with Luke Alfie and his statement that
I can find it.
Distance for an effector mechanism essentially to function. Engineering malaria resistance. So this engineered trait to make your mosquito resistant to malaria, it has to be linked to the gene drive. So we're talking already about two components that these replacement mosquitoes have. They have this engineered trait that makes them resistant to malaria or not being able to transmit malaria, and they have this gene drive component, which can mobilize the resistance trait and move it in the population. So usually when we think of such drives, we think of putting both of these components together, right? In a single place in the genome, both of them are moving together. And this creates the problems that you outlined before. So essentially you have to regulate if you want to test the capacity of the mosquitoes to resist to malaria, not being able to transmit malaria, you have to release or test the gene drive at the same time. And this creates a huge regulatory burden, but also logistical burden and capacity building to do that. So instead of doing that, what we've decided from the beginning is to separate the two. Some people may call it a split drive. You will hear this version. It's not really a split drive. It's somewhat different than a split drive. But anyway, I mean, we can call it like that if you wish. So essentially the two components, this trait, malaria-resistant trait, and the gene drive are in two separate mosquitoes. So we can test the mosquito that has the malaria resistance first, but this mosquito, even if it's released in the field by accidentally or whatever, it cannot move because it has some fitness causes we analyzed before. So it cannot move, it cannot persist there. It will disappear within a couple of generations, it will disappear. And it is only when the two mosquitoes are brought together that the drive will mobilize the effector and they will both start to move. So that's the whole idea of this integral gene drive that we present here, that you bring these two together and this will be done at the very end of the process, essentially, when you are ready to go, when you have established that this is fine, is working, it would benefit the communities, it has social public acceptance, etc., etc. Only then you will release both of them at the same time.
And you do not think that delaying that process does not compromise the fitness of the mosquito? So you're guaranteeing that the mosquito will be fit anyway when you eventually merge the drive and the effector?
No, I mean, you will release them both at the same time. So you will test the first one, and then you will test the second one separately, and then you release them at the same time. So it will be the same quality controlled mosquito, essentially, that you will be releasing at the end.
Oh, okay. So it means that the separation is until release. So even during the release, you are releasing two separate products.
Yes, exactly.
Oh, okay. All right. Yeah. So again, thanks for explaining the technicality here. But we see the reason that you don't want the.
So, for example, now, I mean, you asked the question, Fredros. For example,
Yeah.
now we have generate the fed transgenic mosquitoes in Tanzania. We are in the process of generating the malaria-resistant mosquitoes in Tanzania. Maybe in NIHI, maybe our colleagues, my colleagues there, will be can talk about that, Dixon and Tip. And we didn't have to face the regulatory burden or process that to generate gene-drive mosquitoes. So we are testing them in contained facilities in Tanzania at the moment, but we are not using gene drive. But we are the final product, essentially, this malaria resistance without using the gene drive.
Excellent. Tib, Dixon, do you guys want to say more about this approach to shortening the regulatory pathway?
Yeah, I guess there is a sense of promises and confidence in the approach itself, but it doesn't mean it won't still be questioned in terms of legalness on regulatory aspect. But as George pointed out, when we are requesting the approval from the competent authority to do this work, it was so your intention is at the end genetic gene drive technology, but we're saying no, at this stage we are testing different part of the technology, but at the end we'll of course have a complete set of the technology itself. But the beauty is each step is informing on what you have achieved, and it's creating awareness within the regulatory system itself. So at the end, when they are evaluating the complete set, at least they're already informed on what it entails. I guess it would have been different if it would have to be take all this at the beginning for them to start questioning of what it means. But the beauty is, and it's my expectation that in the next 3 or 4 years, when you go back to the competent authority for the permit, they will already have substantial knowledge on what we're working on, and they understand it's not a new thing, but it's an additional layer of what we intend to develop at the end. Thank you. Back to you.
Yeah. So essentially, Dixon, that's great.
Excellent explanation. Excellent explanation. Go ahead. Yeah.
Essentially, you will be adding to the dossier. Essentially, you won't be bringing something new. You will be adding to the dossier that you have already discussed with the regulators in the past.
Yes, that's true.
No, thank you guys so much for that. Again, if anybody has any question to our colleagues here, please raise your hands and ask. Too much technical detail. I would like us to take a breather from the technical detail. My colleague Lena is live. She's been very active on the chat, pushing a lot of questions, and I think I'm going to let her ask the relevant questions here. But she has a question of her own. First of all, Lena, go ahead, please.
Yes, to George, this is very interesting. I just now realized that 2 different products will be released. So my question is, if you release the drive separately, are there any plans to ensure that it doesn't, without the transmission blocking, that you're just not going to drive it, this new strain of mosquitoes? Like, how do you ensure
like a cargoless drive?
Yes, exactly. How do you avoid that?
Well, you can avoid that. I mean, that's so the current design is that the drive can drive anyway. It can drive itself anyway. I mean, we have thoughts of how to possibly engineer it further so that it cannot drive until the effector is already there. It's also there, but this will compromise the capacity of the widespread drive. So we have to balance those things, but we are not concerned with that so much, Lena. Thank you for the question, because we will be releasing both at the same time, and if we see that the effector cannot keep up with the drive, we will release more effectors essentially. So it might not be a single release; it might be stage releases to keep up with the effector. Now, having said that, the drive itself has its own fitness cost. So it might be also disappearing, but it will be mobilizing the effector, which has less. I mean, let's not go to the technical details. So you can balance the fitness and the capacity to drive. So you can make sure that both of them are driving at the same time.
Sorry, just one last. Why would you not combine them first and then release the final product? Because
they could separate again. So that's that's integral to the integral gene drive design. So we have the effector and the drive and the Cas9, essentially the CRISPR component, inserted within endogenous loci in the genome, endogenous genes in the genome. And these genes are separate. They are in separate areas in the genome. So they will separate anyway if you put them together. But there is but I mean, they may separate anyway. I mean, not necessarily. But there is this option as well that you can put them together. I mean, I'm not saying no. I mean, you could put them together already in the lab and then release one mosquito rather than two.
All right. Let's proceed. Thanks a lot for that. We will try and move faster. We're past midway now. I hope our speakers don't all drop out. Please stay with us. Kindly stay with us. It's very a few more exciting questions ahead here. Ethics and equity and communication. How do we communicate all this technical detail? In one paper we read, we see this, which my colleague Lena has seen a lot also in the conversations in the program we do, African Conversations, where people are more likely to elevate the risk about gene drives. When you start talking about gene drives or genetically modified mosquitoes, people are more likely to talk to you about the likelihood of things going wrong as opposed to the problem it can solve. And I think Damaris was saying earlier that when you speak to the mothers or people in the rural areas, they really need help. But when you speak to professionals, they have a lot of questions. The other thing we see here is that the benefit of eradicating malaria is rarely spoken about. And there is now, we do not take adequately into account what would happen if malaria didn't exist, the economic benefit, the health benefit, etc., etc. And I guess for us, the question then is, to our gene developers and also to our colleagues from the countries, and anybody really from the countries, feel free to. Today we're doing an open masterclass, so anybody from the countries kindly jump in and speak. What would be the best approach to communicate the subject of gene drives as a malaria control intervention to the different stakeholders in the countries? What should we be saying here? At least as professionals, what do you think we should be saying? So anybody wants to respond to this? Anyone from Nigeria, Uganda? Our colleagues in Auda, Nepal, please, come on, help us here.
Yeah, Fredros, maybe I can say something. This is Jesse Gitaka from Kenya.
Oh, go ahead, Jesse. Nice to meet you.
Yeah, thank you, Fredros. Yeah, this is really interesting, and I think the question on communication is, of course, a key issue. As we all know, communicating science sometimes is not so easy. But I think it will be critical to start engaging communication professionals from these development stages to sort of help them, empower them to develop the language that can be easily understood by the communities and even policymakers. I mean, you should not leave them behind. So yeah. And so I think it's a matter of multidisciplinarity. We have to work with local communicators, even the language itself. What is the Swahili word for that? What is the Luo word for that? What is the Gikoi word for that? And what is how the concept of tinkering with genes and then leading to population reduction of mosquitoes? How do we put that in culturally sensitive language and easily understood language? So I think it just calls for multidisciplinarity as well. Thank you.
No, thank you so much. Thank you a lot, Jesse. Yeah, anybody wants to add on to the multidisciplinarity issue here? Actually, my colleague Prashant is still on the call. Prashant, the modeling part is even more complicated than the genomics part. How do we explain these things to people, my friend?
Putting me on the spot.
You know, I
Yeah,
remember when you first came to Ifakara, my colleagues were saying, "Oh, that's the guy who draws cobwebs." Because you see, when you draw your modeling graphs, they look like cobwebs, and so
yeah.
people just know you as the cobweb guy.
Yeah.
But how do you communicate this to?
Yeah, I think it's a complicated issue, but I feel like with modeling, I guess it helps you understand what the outcomes may be, you know, kind of looking ahead. And I think that is the major self of modeling, apart from like how useful it can be for developers and kind of understanding where the field is going. But I think at the end of the day, like it's just a small component, right, of a much larger much larger like picture. So it can be useful, I think, with some of the imagery and like some of the predictions to kind of help people understand. But I think most of the work's going to fall on kind of, as Jesse was saying earlier, like community engagement at a more basic level, you know?
Yeah. Yeah. Do you think that we need to maybe ask my colleague Lena here? Lena, you've done a lot of work with stakeholders across Africa. Do you think we need to first of all ensure that the professionals or the people really at the front line sufficiently understand the technology? Lena, back to you.
Really, yes, that's necessary, of course, and understand it in lay terms. But that's still difficult, even in this discussion. I was just interested to hear that it's still relatively hard to explain this technology in lay terms. I think one person, when I was doing the discussions in Kenya, said, would I be able to explain it to my grandmother, and will she understand it the way that I want her to understand? So it is difficult, but it's not undoable. I think one recommendation that we got was to not necessarily, I know there have been calls to maybe sit with linguists and design sort of user-friendly language around this technology. But the recommendation was, instead of sitting with linguists, sit with people and use the everyday language that people use instead of coming up with new language for this technology. So in terms of how we communicate it, I think we need to talk, as you know, Fredros, we've gone to the community and explained it, and in return the community members said, oh, we already do that, and they were talking about selective breeding or using hybrid crops. So the concept itself is not necessarily new in the community, but how now to differentiate from that community knowledge and say this is a little bit different than selective breeding of cows or chickens. This is, you're adding something else, you're making sure that the gene actually spreads. So finding friendly words to do that is new. I've been asking this question to everyone that I've spoken with. How should we find what are sort of user-friendly terminologies for this? And I'm yet to come up with any.
Yeah, maybe we should hold here a little bit and just ask the audience if you can talk, that would be fine, but you can also just put it in the chat. We really need help with this. How do we communicate gene drives to people? Again, just raise your hands or just start speaking. I think this is really important, and it's a difficult subject. So anybody who can help, especially from the countries or even the people from not from endemic countries, please help us. How do we communicate gene drives? Anybody willing? Let me see if I have friends in the audience. Oh, Abraham, please help us here. What do we tell people? I am too scared to move forward from this question because it's really, really important one for us. Professor Abraham, go ahead. Yao Afrane, my colleague from Ghana, kindly help us here. What should we say? Go ahead, Abraham. Hey, Dixon, what are you guys telling people in the villages? Nobody wants to talk.
Maybe Fredros.
Go ahead, Damaris.
I can give it a try.
I think Richard wanted to say something as well. Richard, did you want to say something? And then Damaris will go.
Okay.
Go ahead, Damaris. I will comment afterwards.
All right. Okay. Damaris, go ahead.
Yeah, I'm just trying to do, it's not guesswork, but what I think should be appropriate, what we should be telling them. And for me, it's what it is. What is mainly, what is a gene drive? What is that mosquito that has a drive, and how does it differ from the wild mosquitoes or the other mosquitoes that have been there? How does it differ from other technologies, sterile male techniques and other STI? So for me, it's telling the community what this exactly is and what it will do. And just in case things go AY, how will we be able to counter or control that? So bringing it down to the basic layman's language, that's what is needed. The other component, I feel that, yes, there are different stakeholders we are talking about, is the kind of tools that we use to reach these stakeholders. I've been seeing that as we leave a lot journalists out when we are doing this kind of communication. So bringing journalists on board, because they know how to speak to the various stakeholders when they are presenting information at community levels. They know how it should be packaged. So I feel that we need to bring in media on board strongly as the key stakeholders to help us communicate.
Thank you. Thank you so much, Damaris. Bring in the media. Richard, please.
Yeah, Fredros. Yeah, well, I agree with that. I just caution that, the media can be in some ways a double-edged sword, especially media outlets have a sort of anti-GM agenda. But in my experience with transgenic Aedes aegypti, I definitely say that widespread acceptance of, is needed amongst stakeholders, especially the local public. Otherwise, it's got potential to become a political hot potato, really, in such countries, even at the evaluation phase. So, how to communicate and the messaging, I would really say that, that the messaging has to be devised, together with professional communication companies. That would be my advice.
Thank you. Thank you. But you agree with us that it's an important thing that we need to configure, right?
Absolutely, extremely important. That's my experience. I made a comment in the chat about this as well.
No, thanks a lot. Barbara, are you able to unmute now? Barbara, if you're able to unmute, go ahead. Otherwise, Dixon, please go.
Yes, what I wanted to say, I think communication is a huge pillar within the technology itself. But again, there's so many things that will drive it. One is, what is the existing experience on the ground with regard to the burden you're trying to address? Apart from that is also, what are the preliminary information that we have in terms of developing the technology itself? I think if we have the results that we have seen in the lab, or whether in the cage, which are extremely promising, it will also guide us in terms of how best we can communicate. I think at the stage when you ask, what do I speak in the villages, there's so much unknown. What we know, even at high level of stakeholders engagement, what we speak is this is what was done at this stage, but we cannot promise that what we will do at the end of the spectrum. Of course, we have hypotheses, but all these things depends on what will happen at the end. So my take is the message will depend on the results that we generate, but also depends on the context of the community that we're interacting with.
Excellent. No, thank you guys so much. Let's move forward now. I would really appreciate that. But if anybody still has comments on how we could communicate this better, kindly add that in the chat. We're going to move to the next topic now, which is insecticide resistance, not gene drive resistance, but rather insecticide resistance. And this is an important one as well. I guess for context, most of the malaria endemic sub-Saharan Africa has pyrethroid resistance. We are going to be releasing mosquitoes with gene drives. The suggestion that we have had from the developers is that they want to go into these communities, collect the mosquitoes from there, put the construct in the mosquitoes and re-release them, essentially meaning that they will be releasing back pyrethroid resistant mosquitoes, or maybe not, I don't know. A question for us here is, how do you think insecticide resistance will influence gene drive programs across Africa? And I would love to start here with our colleagues from Nigeria, if they're still there, or from Mozambique, Dulce Saria, if you're on the line, if you could help with this question, that would be great. And then I would like us to also hear from our gene drive developers again what their thoughts are. So let's go that way. So either Damaris or Basiliana, and then Dulce Saria. You guys there? Just start speaking. Go ahead.
Basiliana, would you like to go ahead, Basiliana?
I think it's on you, Damaris. Go ahead. Hey, go ahead, Damaris. You're muted.
Yeah, I'd wanted Basiliana to go ahead because I think she's been somehow on this a little bit. Then I can come next. I don't know if she's there. If not, then I can get us started.
Right. Basiliana, are you there? Maybe not. You can go ahead, Damaris, and then we will have Dulce Saria.
So I think community, I think Fredros, you asked around community engagement, right?
No, about insecticide resistance. Sorry about that. How would insecticide resistance impact efforts to release gene drives?
Yeah, I think for me, what I look at specifically, since most countries now are reporting insecticide resistance to most of the insecticides that are used in country, such as pyrethroids. So for me, I look at the gene drive, how when it comes out, is it going to be also these mosquitoes are they going to be susceptible to the pyrethroids that are being used, or are they going to be resistant to it? So that's the most important component to look at, because if you are going to implement it along with the other control tools, then that's an important factor to look at. But if it's going to be standalone, assuming like I look at gene drive being implemented in the highlands where malaria is endemic and you don't do IRS or you don't have insecticide treated nets, so then that maybe insecticide resistance could be a question. However, since we are saying that this tool is going to be considered or be rolled out together with other control interventions, it's important to think critically the impact of insecticide resistance. And if the mosquitoes with the drive are going to be susceptible to the insecticides, then it means then you release them and then maybe they are knocked out. So you won't be having a population, good sample size that will be able to propagate in the community and counter malaria infections. Therefore, they are going to be susceptible to the insecticide, and then malaria transmission go ahead. But if then they are not affected, or there's no effect from the insecticides, then that will be. So it's important for that to be considered at this moment before rollout.
Excellent, excellent. My colleague Prashant has done some modeling work on this subject. Prashant, I'm putting you back on the spot, if you don't mind.
Go ahead.
So you and your colleagues have worked on this a little bit from an in silico angle. And let me let you explain what do you find? How does resistance come into play here? Damaris is very categorical here that, you know, we have to choose whether these mosquitoes are going to be susceptible or resistant, and that they will be affected differentially. What do your models say?
Yeah, so we haven't looked at insecticide resistance and gene drive releases at the same time. So we've looked at gene drive resistance, you know, but not insecticide
This is your
resistance.
paper, no? Insecticide
Yes.
resistance.
Yeah, so
Gene drives.
this paper is talking about how you can, with the same model, you can look at both. So essentially that first figure there, the colorful figure with the male and female mosquito, that's just showing how mating happens within the model. And then you can get these offspring that can have different kinds of phenotypic traits. So we could look at it with insecticide resistance and gene drives using this model, but we haven't so far. Yeah, I think what you release, whether it's going to be resistant or susceptible, I think from a gene drive perspective, it wouldn't matter so much. I think it's more like the, you know, how are you going to sell it to people? And then the other thing is, yeah, I mean, for the insecticide resistance component, I think that's something you want to think about as well. Tony's got his hand up. Do you want to jump in, Tony?
Go ahead, Tony.
Yeah, I mean, without pretending to be a modeler, so you know, this has to be qualified a little bit.
Yeah, that
think it's
applies to me as well,
to-
because I clearly misunderstood Prashant's paper. I thought he was actually making a conclusion about resistance here. The model can be used to understand the rise of resistance.
I think he's saying that the same type of modeling could be used to
Can be used for that, yeah.
Could be
Go
used
ahead,
to
Tony.
model the impact of all those three different things.
Okay.
You know, I think it's important to remember how the gene drive works, though, because the gene drive is released on a certain genetic background. But let's say you have a scenario whereby you release a susceptible mosquito. You release, I don't know how many it might be, 20,000, 50,000 mosquitoes. As long as some of those mate with the local wild type population, assuming that that is insecticide resistant, then that gene drive in the offspring is immediately starting to become on the genetic background of insecticide resistance. And so the impact that it will have is largely in the very first stages, where you might have an expected frequency in the first generation of 20%, it might only be 2%.
Right.
Because it has this invasive potential and exponential increase once it's in the genetic background locally, it shouldn't make too much difference. Again, I'm not a modeler, but that is the general prediction. And I'm pretty sure that's what will be borne out by the modeling, more or less.
You know, I'm wondering, Tony, while listening to you, that whether it would be actually good. I mean, I'm not saying that we should do that, but whether it would be good actually to release mosquitoes that are insecticide resistant already in an area where IRS and bed nets are widely.
Sure, and that is a concept, subject of discussion at the moment already. You know,
yeah.
But what would be the advantage of doing that? Because I mean, current modeling with susceptible mosquitoes, that there is a synergistic effect. What would the point of releasing resistant mosquitoes be?
Or that they won't be, yeah, but I mean, these initial releases, I mean, if you're planning a couple of initial releases, or I mean, some in a short period of time, and you are competing with IRS in this specific initial release, essentially, whether it would be advantageous to have them being resistant already.
Yeah, I should say also, like the interventions are targeted mostly towards female mosquitoes, right? So far, at least ITNs and IRS. And releasing male mosquitoes kind of, you know, will still keep your gene drive alive. So that's like, yeah, there's, all I'm saying is like, you can release susceptible mosquitoes. I mean, when you talk about susceptibility, I guess you're specifically referring to females here, but if you're doing male releases, it doesn't matter so much. And with females as well.
Most of the interventions that we do, yeah, that's true.
Exactly, yeah.
That's an interesting perspective there. That so, Prashant, is that on the assumption that the males will not go into contact with the insecticides?
Well, at least the currently deployed interventions are mostly female targeted, right? With ATSBs, maybe you'll get male targeting as well. And there might be some other outdoor interventions that could target males. But so far, it's mostly been female targeting, and there hasn't been an impact. And I should say, like, even if you were just releasing male mosquitoes, and at the start, like as Tony was saying, the frequency of the gene drive in the female population is going to be low as well, right? Hopefully it'll be 50-50, but it's going to be quite low. And even when we're doing that, so when we have the susceptible female mosquitoes, we're still seeing a synergistic effect in the model. In the real world, we may lose more mosquitoes than we do in the model when we do the actual releases. So it'll be interesting to see how that plays out. But you could always mitigate those kind of scenarios by doing more releases, right? Or bigger releases. So yeah.
Good. So at the moment, the assumption is that for these low threshold gene drives, you will be releasing hundreds, not maybe thousands, but not like hundreds of thousands. So
yeah, with the low threshold ones, yeah, it's going to be orders of magnitude smaller release size that's sufficient. You also want to think about spatially how you're releasing, how the populations are mixing, and so on. Because a lot of the models are on single node. There's only a handful of models that are doing spatial simulations. And so when you do spatial simulations, I guess if you increase the frequency spatially as well, that kind of gets your gene drive frequency up much faster.
Do you guys want to talk about that? We can jump to that straight and then come back to the public engagement question. So we talk about the geography concerns, because we have some questions on seasonality and geography as well. And I promise you, Prashant, in this case, we've read your and understood your models. So, but let's start with the S-Notes work. So, you can help us explain this. So first of all, it seems like everything is going fine, but then the same problem that you have with gene drive resistance, you could actually induce that same problem if you release at the wrong time of the year. Is that right?
When you say wrong time of the year,
or if you release at the wrong place or the wrong time of the year. So in terms of missing being inaccurate in terms of spatial assignment, and also the right time of the year to release would give you the same cost almost as if you had gene drive resistance, right?
Yeah, potentially. You're saying that seasonality and like how you release spatially. Yeah, potentially. I have read Ace's paper, but it's been a while, so I'm not going to explain what's on here without looking at it again.
Maybe Ace is on the call. I don't know. Is Ace here? Maybe I'll check.
But yeah, while you're checking, it is. Yeah, again, like you could have a small release size. You could have a pretty spatially specific release, and then depending on geographical features, it may not spread as widely. But this can again be mitigated by larger release sizes, more frequent releases, and so on. So I think the seasonality is important, but especially with low threshold gene drives, I think the impact of seasonality is going to be more on how quickly do you get there, get to the kind of the frequency that you want.
So you're proposing I mean, this is what other people seem to be agreeing with as well, that the best release point is just before the rainy season, right?
Ideally, if you could do you want to increase the chances that your drive will spread. The low threshold ones, again, unless you kill off most of them, they are going to spread eventually. It's just how long are they going to take to establish themselves in the population. But yeah, if you want to maximize your chances of spread, then releasing just before the rainy season, especially if you can do like a small release size, which is relative to the wild population, is still quite large. That would be ideal.
So there is some work that suggests, and maybe our target malaria colleagues can add to that as well, that with the suppression drives, depending on where you release them and the time of the year when you release them, they will not necessarily reach zero, and that you might have some form of oscillation. Can you explain how that happens?
is a target malaria person. Does they want it?
Well, I'm not target malaria. I used to work for them, and still.
Yeah. So what's the idea here? Your intention is not to go completely to zero.
Oh, so what happens? So what you can have is this. I mean, without going into detail and not being able to explain it all, it's called chasing dynamics. So if you imagine, you suppress an area, and let's say you locally eliminate an area, there's no mosquitoes. If there's no mosquitoes, there's no gene drive construct either there. So that cleared area can eventually be repopulated if there's a neighboring wild type population, and then re-invaded by the gene drive as it is receptive. And so you get a suppression and a rebounce and then suppression. And that can happen under some dynamics, and that's why you see some oscillation sometimes.
All right. Okay. All right. So just to close this, back to you, Prashant, and the rest. So in terms of timing, assuming you have a perfect drive, a low threshold drive, if you were to release in the middle of the dry season, because maybe it's feasible, can you counter that challenge by releasing more? I mean, logistically, these things cannot be, we can say we all want to release it just before the rainy season, but in terms of supply chain in real life, it might happen that there are some delays. So wouldn't it be better to like start earlier and just release a little more?
Yeah, logistically speaking, also, I guess getting to different locations in the dry season is going to be a lot easier than, when you're in the peak season. Again, like from the models, and they might be a little optimistic. The seasonality doesn't matter as much when you're doing the low threshold small releases. I guess it really comes into effect when you're looking at like kind of an inundation release, or you're looking to release in a very, very small location, hoping it'll spread to a larger area later. So it's I guess it depends on the kind of gene drive and kind of what your geography is. It's a hard one to answer broadly.
Right.
As I've said multiple times for multiple questions.
No, that's fine. George, just so we can close this chapter and move to the next one. In terms of the rearing needs, what is the size of the payload we're looking at here? Do we need to have mass rearing, for example, or do we just need regular insectry? How do you think this is going to work in practice?
Well, maybe that's an answer for Prashant to give. We're certainly hoping that with the replacement strategy, the rearing would not be that big, essentially, of an endeavor. But I mean, it will all depend on the place, the geography, the mosquito population size, and all that. So maybe Prashant can tell us a bit more of that.
Yeah, I think it depends on the gene drive strategy. And so like Tony was saying, when you get these chasing dynamics, that's another situation where you'd want to consider multiple releases, right? Just so that you stamp out that that those populations that are surviving. With replacement, you also see some chasing dynamics, but it's usually happening at a higher frequency. And the other thing is, because the focus is on the parasite, if you're just trying to reduce malaria transmission, you can still get to that threshold where you have enough frequency in the population that malaria transmission is kind of suppressed. You don't need to get 100%. Same is true with suppression as well. I guess you don't need to kill all of the mosquitoes. If you get low enough, they will just die on their own. But it's a slightly different metric when you're thinking about low frequency versus high frequency. So yeah, depending on the gene drive, I guess you may have to do more releases or not. I think with replacement drives, is, as George was saying, you're probably going to look at like much smaller rearing facilities. I think you should be able to get away with it.
But certainly, you do need rearing facilities in the place where you will be doing your trials or releases. So
but
that
it's going to be a logistics thing, like getting the mosquitoes, to the actual police side.
So maybe Frederick is still on the line here. I mean, a common suggestion that we have had is that countries should have a central rearing location to ensure quality assurance, and then distribute eggs so that in the satellite sites, people can just raise the eggs to adult stage and then release those instead of having a gene drive insectary in every district. What do you guys think of this approach? Frederick, I don't know if you're still in the call. Frederick,
yeah, I'm still there.
Yeah.
Can you hear me?
When you guys think about these releases, what are the plans here for how does it look like?
Well, at the moment, we're not yet thinking about continent-wide logistics, as you know.
Oh, let's talk district-wide logistics.
We're thinking about the first demonstration of efficacy, which already are quite a complex discussion. But yes, one vision that's one I share is that you could have some hubs that specialize in production of eggs from different populations from different places, and then those eggs would be distributed to egg rearing units locally, and that would allow, as you say, having very safe and very performant infrastructures, centers of production that would take on the mass rearing, because even though the selling point of this technology is that we don't need a lot of mosquitoes locally to kickstart the spread, if you think about the size of the African continent, it's still a huge endeavor, and there's going to be a lot of mosquitoes produced. So you'd have those production centers.
Yeah.
But then you need, of course, to ship those mosquitoes by plane or to the locally, like to adult rearing units. So there's lots of things to think about. But I think first of all we need to think about the first demonstrations, proof of principles, and do that right. And then, of course, then we'll be in a good position to think about large-scale applications.
Okay. Don't think too far ahead.
One thing at a time, Frederick.
One thing at a time. One thing at a time.
But these are important questions, Arthur. I mean, you guys know better than me, but anopheline is like a little more sensitive to transport and stuff like that than say Aedes mosquitoes, right? So yeah, as George is saying, you probably want to have your rearing centers very close to where the release sites are to mitigate these kinds of issues. Yeah.
But we can dream, and we can also learn from other examples. So if you look at some of the fruit fly control programs, you have those large centralized production centers, and then the flies are distributed where there's a problem.
Right.
So it's a bit the model you're discussing.
We're trying to move from one room of the sector to another, and we always have a period of overlap. Because I mean, moving from one environment, I mean, the same building, the same rooms. I mean, moving from one room to another, your colonies, you may lose them, and you want to have this overlap. So imagine what's going to happen here in totally different environments.
Yeah. And Lena is going to ask you to put the mosquitoes together first before you release them.
Yes.
So we have one last question on this modeling side of things. When we speak to countries, one of the questions that come up is, okay, if I replace, or if I do this, after how long should I expect the gain? So the current interventions, if I distribute bed nets, people see the benefit immediately, or indoor residual spraying, people see the benefit immediately. What is the situation with gene drives here? Is the benefit immediate? Does it take a while? From some of this modeling work, we see that you are reaching saturation after a year or so, in some cases up to 4 years, maybe longer. But perhaps the epidemiological benefit is already visible before that. What do you guys think? I guess this is an important question for timing, right? For planning.
Yeah. So yeah, I could take that to start off with, at least so as Austin was saying earlier, explaining the generation. So you get to say 12 generations in a year. Depending on how large your release size is, that's still going to be a very small proportion of the population at the end of that year. So I would say in some cases, depending on release size, even a year may be too soon to start seeing impact. But then this is, it's like there's like 10, 15 variables in play here, right? And the important ones are, I guess the most important one is what kind of gene driver you're releasing. And then you can start to think about like where you're releasing, when you're releasing, and so on.
Right.
Especially with the low threshold ones, I think you can get to higher frequency faster with bigger releases, as I've said earlier, and doing like a more dense release spatially. But yeah, I think it's just giving those generations to kind of start building up. When in some cases you want homozygous mosquitoes before you have an effect, and so for homozygosity to take over, I think that takes a little more time as well. So yeah, it depends on the construct, and I think we can use these models to kind of predict how quickly that could happen. Helpful tool for communication there. But yeah, there's a lot more to understand about gene drugs before we get to their modeling.
But at least what you guys are saying is that the benefit is not within that year. It's not immediate. It's unlikely to be immediate.
Unlikely, yeah, unlikely with something where you're waiting for the population to build up in frequency. And even like with replacement and with suppression, even with 50%, you will see an effect, but not that much. 50% frequency in the population.
And it will also depend very much on the reproductive rate in the region. So if it's too high, I mean, you have 20 and 30 are not, then you won't be seeing an effect immediately. I mean, you need some time to see that. So I mean, it would depend on the place, endemicity, and everything.
All right. No, thank you, guys. Lena, did you have a question here, or shall we proceed?
No, I'm good.
All right. Okay. So let's go to the last bit of the conversation today. It's been fantastic having all of you here. The last bit of the conversation is really about the multiplicity of vectors. The second last bit, we still have the regulatory side, but the second last bit, the multiplicity of malaria vectors. At the moment, most of the gene-derived products are targeted at Anopheles gambiae. Most of Africa has more than just Anopheles gambiae. The four main ones are Anopheles gambiae, Anopheles funestus, Anopheles arabiensis, and Anopheles colutzei. And I think that the assumption at the moment, and this is from Tony's 2020 publication, that it's going to be fairly easy to transmit or to transfer this technology between species. There might be some challenges here and there, but there seems to be proof of principle already that this should be easy to do. Now, given the vectors that we have, where are we with this transferability between species? And I think in the beginning of the call, we were saying this is a species-specific technique. So if you have more than one species, you're going to need at least two products. So do you guys want to talk a little bit about how this is going to work, or how you foresee this as working as the developers? And then it would be nice also to hear from our country colleagues also what they would expect to see, given that the circumstances that we have there.
Just to add another component, Fredros, to what you said, I mean, this is related to what you asked previously, right? So in a place where malaria is not transmitted exclusively or even predominantly by Anopheles gambiae, I mean, the impact will not be seen, at least not immediately. So it's related to that, just to flag this up as well.
Okay. Frederick, you are our resident entomologist.
Really? That's good to hear. All right, thanks a lot. Okay, so
for the masterclass,
you and some others are acutely aware that some of those species you've listed there are hard to maintain, and therefore there's going to be technical limitations to which ones we can transform. So if we go beyond the fact that we might want to transform Gambier, Coludia, and Arabiensis because they're the main vectors, for example, in West Africa, which I think we will have and we can, we will start hitting some technical bottlenecks with species that are harder to transform and to maintain and to breed en masse. So I think there's no easy solution at the moment. But one thing I was thinking recently is that if we really manage to convince and to demonstrate that these tools are really what they are in terms of cost effective and working outdoors, indoors, in any setting, maybe by that time we will have less safety studies to do for the regulators, and we may be able to explore easier ways of transforming those hard, difficult species that remain. Okay. And by that, I mean, for example, you could imagine that everybody is really sold with regard to the gene drive technology, and we have a mechanism of effector genes that are well known in those first 3 species we target. You could maybe have a system where you catch Anopheles melas, you transform it, and you release it. You would skip the maintenance and rearing bottlenecks, if you see what I mean. That's very easy to say, but I think for some of those very hard and minors or secondary vector species, hopefully we'll have transformation strategies that are a bit like that, that are less intensive.
Okay. Tony, I see your hand.
Yeah, I think there are a few things to say here. One, you know, we can adapt these technologies to other mosquitoes. We've done, you know, we've done Phonestus now as well. So those bottlenecks are surpassable. But you know, if you look at malaria control today, it has focused on a handful of mosquito species, really, because these interventions like bed nets, you know better than me, and Lena mentioned it also. You know, they are targeting only a certain few species, predominantly of vector, and in those highlighted ones there, and then they've had the most effect on malaria transmission. So, you know, there seems to be a premise that we need to go after every potential vector of malaria, no matter how minor it might be. And I don't think that's either proven or accepted or even predominant prevailing opinion. You know, Lena mentioned it there, where you have caused the species bias in one country due to a specific intervention like bed nets. Those gains in malaria reduction have been maintained. So I think it's important to set the expectations accordingly.
Okay. Yeah. Not be too overambitious, if you like.
Yeah. Not overly ambitious where it's not necessary.
Okay. All right. Let me ask Damaris and Dulcesaria. I think that it's Tony it's Frederick who mentioned this. We will need a lot of information here on the efficacy of the drives. From your side, from where you sit, from the countryside, in your opinion, let's talk a little bit about the data that we need, not for implementation, but the data that we will need to allow this technology to be tested in the country. So as a representative of the country, let's assume that for now. Richard, I hope you're still on the call as well. You could help with this. What kind of data should we be asking of the developers to allow us to do this kind of testing, to allow us to evaluate gene drives in the country?
I think maybe from my end, I think it's important, especially for us to be looking at efficacy of this as the data I think that would be of must have is the target. Because we know the mechanisms of resistance where we have both genotypic and phenotypic. So we will need to understand what are those target regions of the drive and how they counter with the target sites where we are monitoring insecticide resistance. So how do they contribute to development? Are they in isolation? Does the drive actually contribute to mosquito developing resistance easily to the insecticide? So we'll need to have to understand the target side, but also the other components that we'll need to understand as far as resistance is concerned is to do with species, how differential this, how the insecticide that we're using are affecting the drive. Insecticide that will be, because there are all lots. And here we shouldn't be thinking about just the malaria control insecticides. There's the ones that are used in the agricultural setting that also can contribute to, can affect the gene drive, the mosquitoes with the drive. So I think the data that will be important for us is both genotypically the drive and also phenotypically how these mosquitoes can be affected by the insecticides that are being used out there, both in the fight against malaria, but also in the agricultural sector.
Would you need any information about the construct themselves? So for example, we talked earlier about the integral gene drives, the ones that Transmission Zero are working on. What kind of information would you need about this, other than the mosquito itself?
Yeah, it's important to have information about the construct, because again, maybe I'll give an example. Recently, we're doing some resistance monitoring of Culex mosquitoes, and we were subjecting them to various compounds, only to realize that these Culex mosquitoes are having KDR, and it was contributing to the level of the concentration of the insecticide that you needed to use to fight this, while we know that there's not much contribution of KDR to phenotypic resistance. However, we registered this and were able to document that actually there's some slight contribution of this. So we need to understand the construct, but how do we relate the construct with the resistance development? That will be the thing. How does the construct contribute to, how does it affect susceptibility of these mosquitoes? I think that's what I would request.
No, excellent. Thanks a lot. I'm wondering whether we still have Dulcisaria. Dulcisaria, do you have any additional points you want to make here? Lena, your hand is raised as well. Go ahead.
Specific comments, but just questions in this case. In terms of safety and efficacy, for safety, I think I understand that we can get evidence, but for efficacy, I was presenting to my team this morning about sort of the process of looking into what profiles for gene drives we could make for Africa, for example, without any prior knowledge of how, like, we don't have any experience that we can say, oh, we can learn from X country that has done this, when no one has done this at the moment. How can we even put the endpoints that we want? Because this is information that has been requested by all the stakeholders that we have spoken with. They'll say, what are your thresholds or minimum requirements? How well will it work? Or what? How can we set these endpoints at the moment when we don't really have anywhere to learn from, or any similar, or technology that is so similar to this one that we can say we can learn from this? For the experts, do you know, for example, what would be the minimum efficacy for gene drives, for whichever approach that we can respond to when these questions come up?
Well, I guess the simple answer, Lena, is that we don't know yet. I mean, I'm speaking on behalf of Transmission Zero, but I think the same is true for for other programs as well. So, I mean, what you're asking essentially is what we call the target product profile. Essentially, what are the thresholds, the meaning, the requirements? Essentially, what is the what will be the efficacy of that? So, yeah. So the short answer is that we don't know yet. I mean, we may have to establish that, although that's not universally agreed that we need to establish such thresholds. And it may be just enough to say that we need to, I mean, we need to have an epidemiological impact at the end and be a measurable impact. So yeah, I mean, that's more or less the answer. So we don't know yet, but these are things that we should be figuring out. I don't know if Tony has anything more to say on that, but.
Yeah, and if while Tony is preparing, do we have anybody from the countries other than Kenya who has a specific question or a specific request for specific kind of data? Do we have Dr. Shija, for example? Anybody from the countries? It's past.
So Fred was just a small comment here.
Yeah.
So when we've spoken to policymakers, one of the things they've asked is the effect of the gene drives on the disease. Like how long will it take to reduce disease burden?
Yeah.
You know, and of course we can't do an experiment on that right now. We could have probably some modeling data, you know, and more recently, when you speak to decision makers, they also try to ask you something on how that links to development. And of course, there are many ways that it could link to development, but we don't seem to have any real text in the recent past that could explain this kind of stuff. So,
do you mean country development, Richard?
Well, it's a very open question normally. You know, it's not restrictive. Yeah.
Yeah.
There are economic models that one could apply. Once we have a model on the efficacy, there might be financial economic models that macroeconomic models that could apply and could tell us about how it contributes to development, sustainability, etc. in each country. But yeah.
Yeah, and one question that we don't have here today, but which we've discussed with people individually is what components of this technology are monetizable at country level? What role can the private sector play, for example? With technology such as bed nets, you know, you have a lot of job creation, you know, people are distributing nets. Or people have level source management, a lot of job creation. People are spraying houses, spraying habitats. Gene drives is probably not going to be the same. So,
well, it could be further. I mean, we can say we can be very creative there as well. I mean, we have to think post-release monitoring and implement, you know, post-implementation essentially, and the monitoring of all that. So, I guess the national malaria control programs may not be enough to do that, and maybe the private sector could play a role in that.
Oh, that's nice to hear. Yeah. Richard, you wanted to add?
Yeah. So this question comes very much from parliamentarians. They ask directly, is there a chance for us to make money if we approve this technology? You know, that is always a direct question. And I've spoken to gene conveners sometime in the past, and we try to answer this question in terms of the benefits that come when we eliminate the disease, you know. But these guys want us to go a little bit beyond that. Yeah, which might be difficult for this technology.
Yeah,
well, it's difficult, Richard, because these guys usually stay in the parliament for 4 years or 8 years maximum, so they may not be able to reap the benefits of a long-term, you know, development of the country. But that's the answer essentially. I mean, if the country develops because there's less malaria, essentially, then everyone will be making money, or you know, will be developing. So I think that would be my answer. But you know how that works.
Yeah, true.
Right. Let's wrap up. We have the last session. I'm going to run through this real quick, and Damaris then will take. Or do you have a comment on that specific topic? Damaris, go ahead.
No, I just wanted to also say the question that they're asking is, we have to do one molecular biology, but do we really have to do gene drive? This is what the parliamentarians ask, and that's why they're looking at what bullet other than investing heavily on gene drive.
Yeah. Does that mean that if the gene drive investment was designed such that it's serving an additional purpose, it would be much more acceptable?
I think so. Yeah, but maybe Richard and the team could speak to that.
So if you integrate this like molecular surveillance thing to serve additional purposes instead of just being gene drive, okay.
Sure.
Right. No, thank you guys so much for that conversation. The last bit we have is on Prashant, are you still with us?
Yes.
Epidemiology question here. Where do we start? Where do we put gene drives? Target malaria people, UCI, UCMI, transmission zero, they have refused to say whether this technology is for high transmission or low transmission. And I think they have a good reason for not saying. It appears from your models that you have this simulation for high transmission areas and also simulations for low transmission areas, Prashant. And in both cases, it seems to work. There's a little bit of slow activity on the high transmission side, but gene drives still work fairly well, at least the ones that you've modeled. For a start, when you are starting, do you think it matters? How do we fit into this subnational stratification agenda that is very actively being taken up by countries at the moment? This is a map of Tanzania. You see the distribution, the north-south, middle corridor there, very little malaria. How do you think gene drives fit into this context of stratification? And in case we still have my colleagues from Tanzania, it would be nice to hear from you as well on whether you think gene drives should be specifically targeted at low transmission settings or high transmission settings or wherever, or we shouldn't care about those.
Does someone from Tanzania want to go first?
Damaris, is your friend still there?
Who is that? My friend?
From Tanzania.
And Basiliana.
Basiliana.
Basiliana, are you there? I don't know. Maybe she's having network issues.
Anybody from Uganda, Tanzania, Nigeria? We have people here who from these countries. Kindly help us here. Where should we be putting the technology? What would you prefer? Oh, Prashant, you can go ahead.
Yeah, I think it depends on the drive again, and then what the goals are. If you want to see really quick impact, I think releasing gene drives anywhere is going to take a lot longer than say doing an insecticide based intervention. So yeah, I think there are nuances to high and low transmission settings. George touched on this earlier. With the high transmission stuff, you will see a drop in cases. So if you're looking at a percentage reduction, you might see a pretty quick drop in cases potentially, but you have to get to some threshold before you see that. With the low transmission settings, I think you can see that drop a little faster because even a few, a handful of cases that are fewer make a much bigger percentage difference. The problem with low transmission settings, I guess it's been hanging around there because there's importation of malaria and so on. And I don't think any of the models have done a good job so far of doing ACE-NOT was one of the first to tackle it, and they did a reasonably good job, but it's really hard when you have importation of malaria, especially in low transmission settings. And then the other thing is, depending on the gene drive mechanism, which species are kind of sustaining that transmission, right? And so you can't think of gene drives by themselves. I guess you would want to think of it in synergy with other interventions that you're handing out. Yeah.
It's a very complicated question, approach. I mean, the other component of this exercise is also the vector species. Usually, typically, in low transmission settings, Anopheles gambios or Anopheles funestus are not the main vectors. So it would depend very much on that. So if you want to target a low transmission setting, I mean, you possibly won't be doing much of a gene drive in the Anopheles population, right? So you won't be changing much of it. So on the other hand, communication-wise, it's really important to see a fast, a quick, outcome to promote the program. So you have to choose based on models, I guess. You have to choose very carefully where you would do the first tests, so you can have some outcome. I mean, my bet would be that you have to go, because we're talking about Anopheles gambia mostly here, it would be certainly the first essentially to be applied. It has to be a heartland of Anopheles gambia, and it has to be a rather high transmission setting, and we have to measure that as a drop in the malaria cases, not necessarily elimination, eradication, or control even. It has to be a substantial drop, measurable, so that you can communicate that much easier.
Yeah, and then with the background of other interventions also. So that's another thing to keep in mind. And especially with things like ITNs, which are distributed every 3 years, you see these kind of spikes in performance, as soon as you distribute them, you get much better control, and then you see it's starting to wane over the 3-year duration. So that's something to keep in mind as well. Yeah.
Great. So would you expect that in the high transmission area, establishment is quicker than in the low transmission area? I mean, establishment of the drive. Just assuming that the mosquito numbers are low as well, right?
Yeah, I guess it depends on why transmission is low. Is it because the mosquito numbers are low, or is it because there's less like parasites in the region? And I think, depending on that, I could answer like how quickly is it going to establish?
Because you were saying earlier, Prashant, that in the low transmission area it will drop faster.
No, so you could see a significant change percentage-wise, because even a small change in the number of cases, for example, when you already have low transmission happening, you have fewer cases. Percentage-wise, a small drop is going to show up as a bigger change. Whether it's going to be faster or not depends on how quickly the gene drive spread within that population, and then which species we're targeting, as George said.
We have seen that with COVID previously, Fredros. So the R0, the reproductive rate of COVID, was lingering around 1 point something, and 1 and 1.2 for such a long time. We were able to bring it down from 3 point something to 1 within a matter of months. But it was staying there around 1 for such a long time, and we couldn't really go to control. It would be the same with malaria. So what is success? I think. I mean, this is what we have to have in mind. What we should consider a communicated success.
Yeah, that's a good point actually. So when you have high transmission, a lot of times it's just 1 or 2 species that are dominating, right? And if you target those species, you'll see a much faster drop. When you get to the low transmission setting, if you have like a lot of different species that are transmitting, then there is that plurality that you have to deal with. So
right.
Yeah,
we just put up the image of Zanzibar there. They have below 1% for a long time.
And most of it is being imported from the mainland, right?
Correct. Most of it is imported, so you might.
I mean, all of it right now. I think I don't know how else they would get malaria, but I think that becomes like a major driver of transmission. When you
so I guess the question there is, how would gene drives play a role in that?
Yeah, there's human migration as well as vector migration that we have to think about. And there are places like Sri Lanka that are kind of comparable to Zanzibar, because it's an island country, and they took like some very strict like they had some very strict monitoring around malaria cases being imported into Sri Lanka. And I wonder if you'd have to do that in island settings, to kind of control malaria. But what do you do in the mainland then? It becomes a lot harder.
Right.
The question is whether this is sustainable, Prashanth,
Yeah.
economic as well.
Yeah, absolutely.
Richard.
Yeah, Fredus, I'm just imagining or thinking rather that we can try to rephrase your question in terms of to think about seasonal versus perennial transmission rather than low and high transmission. I'm looking at the other tools for malaria control, and if you look in the WHO malaria report, they always talk about seasonal malaria chemoprevention, and everything seems to be looking at either seasonal or perennial, And I believe that if the gene drives are very persistent, like they're going to be present in an area for a very long period of time, they're going to do their job anyway. And that would complement the other tools very nicely. If you think about even the monoclonal antibodies, it looks like they are going to go for that seasonal malaria chemoprevention. And I know that that works very well for the Sahelian states than the rest of Africa, for example. And so even having something, trying to model gene drives around perennial transmission, I think would be a very big story for this tool. Thank you.
Agreed. I actually think that from my reading of S. North's work, there's clearly an impact of the seasonality there, and the extended very long dry seasons have a fairly different impact than short ones.
Right. Okay.
Yeah, and to Richard's point also, I should say, and Ace's work and most of our gene drive modeling, the gene drive modeling has been done by itself. There's usually not inclusion of other interventions. So we don't want to discount that as well. They do have a big impact, even if they deploy seasonally.
Yeah, in Philip's early modeling work, he had very specific, I don't know if, I didn't want to go to that because we didn't include that for the discussion today, but I remember that he had this seasonality built into the model, and you could see very clear differences if you had extended dry seasons. Prashant, you know about that better than us, right?
Yeah, I mean, again, it depends on how high the spike is when there is seasonal transmission, how intense overall transmission is. But yeah, as you can see in his simulations also, there's quite a broad range of outcomes here. For the same kind of, and this is especially true when you get to those kind of edge cases. So calling low transmission an edge case is not really, but there's more variables at play there, and so then you end up with a much broader range of outcomes. Yeah.
Yeah. Anyway, I think we should bring this to a close. I would like to say a big. I'm going to send this back to my colleague Damaris. And I don't know if Dixon is still on the call. It would be nice to hear your voices the last time here. Just before that, the only things that we didn't cover, I just want to mention them for the sake, is the question of regulating gene drives, and I think we will find time in a future masterclass to discuss that in more detail. There was one very interesting subject that we had wanted to talk to you about, which I think is originated from target malaria. Let me see if I find it. This idea of knowledge engagement as opposed to public engagement. So that's something that I would really have loved to discuss with them a little bit more. But I think we preserve this for a different time. I think the idea there is really, really nice and important, the way they're putting it. So that is the second thing. And then there was the issue of code development that I think Transmission Zero has very successfully started doing with the Tanzania work. You could see actually, George, in the integral gene drives manuscript, you still talk about importation. What you have done is far beyond importation. I remember in the pathway document when Stephanie started working on this, they had this stage 0, which was the drives being built in the West. The genetic material construct being built in the West, which was a good assumption at the time. But I think that the path that you guys are taking to try and get things built in the endemic countries is a fantastic step. So it's just interesting that, you know, when you first described integral gene drives, you didn't imagine the outcome.
Yeah,
that
we
you
didn't
ended
imagine
up
that.
with.
Oh, you're right. Actually, we just went with what's currently, what was at that time being thought, and we thought it would be easier to import.
Correct.
But the vision from the beginning was that we will develop it in country. Yeah.
I'll one day when we are sober, I will tell you a nice story, because, you know, when I visited Imperial in 2017, and you said, "Oh, we want to send a container to Tanzania," and I was like, "Oh man, these guys think we have nothing, man. They want to send us a container." But, you know, it turns out that having a prefabricated lab sent to Tanzania was actually a very useful idea, even though we thought it was condescending at the time. Like, oh man, these Imperial guys.
Maybe you should ask Dixon, who will tell you a bit more.
Yeah, they just think that these countries need a lab built, pre-built and sent there. But anyway, it's been a fantastic conversation with you guys. All of you, we didn't talk about containment, but we can talk about that in a different gene drive masterclass. We also didn't talk about anopheles stephensi, how that might impact the work on gene drive in Africa, and we can talk about that at a later time. I would like to stop here, but first of all, really just to say a big thank you for all of you guys, to all of you guys, for all the support that you've given to me and my colleagues at Ifakara Health Institute over the years with these masterclasses. This was the 43rd edition of the masterclasses. We have started to have more and more partners coming in to support, and we hope that we continue to have more of this. So I would like to end by inviting the speakers to say their last words. If you're still on the call, kindly just jump in and tell us your last words, and then we can bid each other goodbye. So I will start with you, Damaris, and then everybody else can follow.
Yeah, thanks a lot, Fredros. I think this has been excellent conversation, and my thinking is that this is the conversations that we need to be having now. I know it's going on with various stakeholders, but if we could be getting one-off discussions like this, because it keeps us thinking and also enlightening on why, because we read papers, but no one synthesizes the information as we've done today. So for me, not only this technology, but other technologies that are being done towards the fight against malaria, it's important for us to keep on the conversation. It's been great, and thanks.
Thank you too. Thanks a lot. Frederick? Did we lose Frederick? Dixon? Tony?
Just thank you for the opportunity. It's always nice to talk about the work and try and get it to a wider audience. I know it's tricky to understand all the details, but you know, we do our best to try and break it down. And my only concern sometimes is that, you know, because it's technically difficult to understand, people seem to automatically imagine that it's difficult to achieve, and it's not necessarily, and it's not necessarily expensive either. So I don't think it should be seen at the detriment of other technologies. So at least this message that it's not a silver bullet and it's complementary is getting across, I think, and I'm happy about that.
Thank you guys. George and Prashant.
Yeah, no, I mean, just to say thank you, Fredros. It was and congratulate you for organizing that. It was great, and thank you for inviting us to share our opinions with people. And thank you to everyone for their contributions in the chat and also the questions. It was
the chat was incredible today. Thank you, guys.
It was a great discussion, and it allowed us to discuss many things, possibly not in enough detail, but I'm sure there will be more to come. So we'll be looking forward to it.
Thank you, guys.
Yeah. Thank you for this masterclass, but also the previous ones. You've been doing a great job. Keeping it's kind of nice to see who's interested and what their backgrounds are, and yeah, the questions have been great in the chat. yeah, I'm hoping like this is just the start of the conversation, you know, to get more people involved.
Yeah, we hope so too. We
thank
hope
you for
that many more
organizing.
people can learn about gene drives and other gene drives in particular, 'cause this is a fairly difficult one. But I'm grateful that, and Lena, you agree with me. I think that our colleagues have tried their best to simplify things for us. So, Elena, do you want to go next? So, is there anybody we have missed who is still on the call?
I have a comment, Fred.
Oh, go ahead. Oh, Richard, go ahead, please.
So I would like to really thank you for this masterclass. I've missed a lot of them. But as we spoke today, I kept imagining that one day you would create a special masterclass where you have a group from the field. You know, think about some community health workers, and they have these views that are very spectacular, things we can never think about. Recently, when Lena did her work in Nairobi, someone came from the slums in Nairobi and asked the question, do you think climate change will affect these gene drives? Gene drive mosquito, for example, you know, just from the wild like that, you know, and it will give the people from the West a very good feeling about how the ground feels like as well. So think about that if it's possible in these master classes. Thank you.
No, great. Okay. No, thanks a lot. Yeah, I hope we didn't miss anybody, but we just want to say thank you all. My colleague Lena, please go next.
Just also wanted to say thank you very much to all our experts. I always learn so much, and it doesn't matter who I talk to. As Richard said, sometimes you'll say, I don't think there is anything that I haven't heard, and then you hear a lot of new things. So I've learned a lot today, and I've enjoyed seeing all the questions and all the explanations. And I just wanted to say thank you very much. I think we'll probably be approaching you again, because we are trying to make sure that we get as many questions answered as we can, so that to avoid any fears that might come up on this. So just we thank you for coming on and explaining this again to us.
Yeah, and with that, I would like to close the masterclass today, but once again say a big thank you to our speakers, and a big thank you to the attendees for staying with us. It's been 3 hours since we started. We finished exactly 3 hours and 3 minutes, and you've all stayed put. We started with 250 people on the line, and I can still see 100 people are still with us. So this is really great. We are grateful for that. The masterclass, of course, is on YouTube as well. As always, if anybody needs the slides or the review material, do let us know, and we will provide that immediately. So this is available for public use. Other than that, we see you next time. With our next masterclass is going to be on severe malaria. And we will find a date for it, a lot to discuss there. And we hope that we will have another masterclass on gene drives as well. This is a big topic, so most definitely we're going to have a second masterclass on that in the coming few months. But otherwise, the 43rd edition of the masterclasses is coming to an end. It's come to an end now. Very grateful to you all, grateful to my colleagues at Ifakara Health Institute for the support, and grateful to my colleague Lena as well in particular, and to all our partners, Gene Convene, FNIH, University of Glasgow, Damaris and PAMCA team, and everybody whom I might not have mentioned. Remember that we love you all. Bye-bye, and see you next time. And
Bye,
Prashant,
thanks.
of course, my friend Prashant, my only Indian friend.
Bye-bye, everyone.
Bye-bye. See you guys.
Bye-bye.
Bye.
Bye. Thank you.
Thank you very much.